Well, good morning, everybody, and welcome to the next in the series of highlighting organic chemistry. And this morning, on the 19th of May, we are celebrating and highlighting organic chemistry in the Czech Republic. So my name is Pat Gyrey, and I'm the president of the division of the EuChemS organic chemistry. And it's a real pleasure to start this morning's meeting and welcome you all. Thank you for registering and attending. And without any further ado, I'll head over to my colleague, Vladimir Sindelar, who's going to tell us lots about the Czech Republic. Vladimir, over to you.
All right, thank you, Pat. I start sharing. Do you see my screen, right?
Yes.
Yep, you just need to go to slideshow, that's all.
Okay, now it's okay?
No. Vladimir, it's better to start slide and then start sharing, because we see.
Okay, let me do it over. Okay.
And don't worry, we always have these slight technical hiccups at the very start of all of these, don't we?
It was before, so I try one more time. All right. Is now better?
No.
No, still the same.
Still the same.
Okay.
So this would be a good time for jokes from the Czech Republic.
All right, let me do this way.
Question is whether someone knows any joke about organic chemistry.
Well, yep, that's always the one, isn't it?
Perfect.
All right, sorry for delay. All right, so good morning again. And first, let me thank the PAT and Division of Organic Chemistry of EuChemS for initiating this great series of online events. And we are very happy today to got this opportunity to highlight organic chemistry within the Czech Republic. So to introduce the chemistry in Czech Republic, I would like to start with the Czech Chemical Society, which is the oldest chemical professional organization in the Czech Republic. It was established already in 1866 as Association for Education in Chemistry and Allied Sciences. So currently there's about 1,300 members. The society owns a journal called Chemické listy. It was established already in 1876, and since then it publishes research papers across all chemical disciplines, and most articles are published actually in Czech and Slovak language. Czech Chemical Society consists of 5 local branches at the different regions. As you see, 5 different cities in the Czech Republic. And it also consists of 27 working groups, which are listed here. So one from these working groups is dealing with organic, bioorganic, and pharmaceutical chemistry. So let me briefly introduce this group. Actually, the main objective of the group is to share information and develop cooperation between the members. So the group organizes a conference called Advances in Organic, Bioorganic and Pharmaceutical Chemistry. The conference takes place every year in early November, and it is a 3 days event with about 150 attendees. The group also gives 2 major awards. Rudolf Lukeš Prize is awarded to chemist for outstanding set of original papers published in the field within the last 5 years, and Alfred Bader Award is for best chemist under the age of 35. There are 2 foundations supporting hugely organic chemistry and related fields in Czech Republic. The first is Alfred Bader Foundation, which provides stipend for talented Czech students so they can do their PhD in prestigious universities abroad. And the second is Experiencia Foundation, which was established by two Czech chemists, Hanna and Dalibor Dvořák, in 2013. And they use their private money to finance several programs. For example, they finance postdoctoral stay of the Czech students abroad, and they also launch start-up grants to motivate young scientists to establish their own research group in Czech institution. So this support is hugely appreciated and has a positive impact on the quality of research and education in Czech Republic. Famous Czech scientist, actually chemist. I selected few of them. Emil Votoček, who is especially noted for his work on the chemistry of saccharides, and he significantly contributed to the creation of Czech chemical nomenclature. Jaroslav Hejrovský is actually the only Czech chemist who received the Nobel Prize. It was for his invention of the polarographic methods. And in 1929, these two gentlemen founded a journal which is called a Collection of Czechoslovak Chemical Communication, which publishes scientific articles. That time it was in English and French. And in 2011, this journal was transformed to Chem plus Chem. I decided to highlight three more Czech organic chemists. Rudolf Lukeš is considered to be founder of synthetic organic chemistry in our country. He is known for his work in the synthesis of keto acids and heterocyclic enamines. And his student was Vladimír Prelog, who received Nobel Prize for Chemistry in 1975. Otakar Červinka is known for his work on stereochemistry, especially asymmetric synthesis, and he is also known for development of mechanisms of organic reactions. And he is the author of a book of organic chemistry, which was appreciated by students and teachers for many decades. And Antonín Holý is mainly known for development of important antiviral drugs used in the treatment of HIV and hepatitis B. So in terms of universities, research and the research institutions, we have 26 public universities, and there are also private universities, but they do not educate in the field of natural sciences. There are more than 300,000 students at the universities, and 21,000 study in the field of natural sciences. From this, 60% are bachelor students, 33% master students, and 7% PhD students. Next to the university, there are also 54 public institutions which are part of the Czech Academy of Sciences. From organic point of view, I mean, from point of view of organic chemistry, you can see here the universities. University of which are mainly contributed in this field. I mean, so this is a Plokyně University, Technical University Liberec, University of Pardubice, Palacký University in Olomouc, UTB in Zlín, Masaryk University in Brno, University of South Bohemia, University of Chemistry and Technology in Prague, IOCB Prague, Institute of Chemical Processes Fundamentals, and Charles University. In terms of funding, the research is supported by Czech Science Foundation, the Technology agency of the Czech Republic, Czech Health Research Council, European Union, and the Ministry of Education, Youth and Sport, and the Ministry of Industry and Trade. In terms of industry, the annual production is about 25 millions, and the largest share of our production is occupied by basic chemicals, followed by rubber and plastic industry. Fourth largest is the pharmaceutical industry, this share of 13%. Annual production is around 25 billions euros, which is about 13% of entire manufacturing industry. Chemical industry employs more than around 140,000 people, which works in 120 bigger company and more than 1,800 smaller companies. In terms of our speakers today, starting from north, is Filip Bureš from University of Pardubice, Lucie Bruliková from Palacký University in Olomouc, Jakub Švenda from Masaryk University in Brno, Michal Kohout from University of Chemistry and Technology in Prague, Hanna Macičková-Cahová from IOCB Prague, and Jan Veselý and Veronika Nováková from Charles University. So that's it from me for now, and I would like now to ask our first speaker, which is Jan Veselý from Faculty of Science, Charles University in Prague, to follow up with his presentation. So please, Jan, you can share your presentation.
Thank you, Vladimir, for the kind introduction, and can you see the presentation?
Yeah, but not in a presenting mode.
Okay. Is it better now?
No.
Still not.
Mm-mm.
So maybe Vladimir, you had this problem. You can explain to.
Yeah, I solved it by sharing the screen, not the file, but the screen. So it worked for me that way. I mean the desktop.
Okay. So is it better now?
Yeah.
Perfect. So sorry for the technical problems, but first of all, I would like to thank organizing committee for giving me the opportunity to have a talk here and to talk about enantioselective synthesis of cyclic compounds using organocatalysis. So I'm coming from Charles University, as Vladimir already said, from the Department of Organic Chemistry, and I would like to shortly introduce what we are interested in. We are interested. Okay, now it's working. We are interested in the development of enantioselective methods for the production of optically pure compounds. And in this area, I can say that we are interested in development of new organocatalysts. For example, we were interested in the synthesis of bifunctional organocatalysts of this type. For example, we combined carbohydrates that are readily available and also amino acids, and from those two units, we were able to synthesize bifunctional catalysts containing thiourea moiety and phosphine part. Also, we were interested in the formation of C2 symmetrical organocatalysts composed of only carbohydrate units, composed of aminosaccharides, and also other systems. Nevertheless, because of lack of time, I would like to just shortly talk about the application of first type of catalyst, and we have applied those catalysts for the production of optically pure allylic type of alcohols through enantioselective Morita-Baylis-Hillman reaction. Second area of our research is the development of enantioselective transformation using small organic molecules. There I have picked up one example, production of enantiomercury pure allylic amines. Those allylic amines were obtained through either first method, when we used classical carbonates, Morita-Baylis-Hillman carbonates as in a racemic form, and as the nucleophiles, various type of amines were used, and under catalysis with tertiary amines, for example, beta-isocupridine, we were able to obtain a high level of enantiomer control of the formed allylic type of amines. The importance of allylic type of amines is not necessary to mention. Nevertheless, there is just shown how we can easily form alpha-alkylidine beta-lactams from those systems, and those beta-lactams are straightforward way for the synthesis of biologically active compounds, including ezetimibe as a cholesterol-lowering agent. The most efficient method for the production of allylic type of amines we have developed is based on atom economical approach using carbamates instead of carbonates. If we take a look to the second transformation in detail, I can move to the reaction pathway that is simplified. We used carbamates that were in situ generated from racemic type of alcohols by the reaction with isothionates. And after formation of those carbamates, we were able, through double SN2 prime process, activate through the decarboxylation corresponding carbamate moiety, and the released amine or amide moiety attacks electron-deficient beta position in an activated Morita-Baylis-Hillman intermediate, and gives chiral type of amines. So this nicely demonstrates that we can apply organocatalysis also for production of biologically active compounds, but we are not only interested in single formation of single bonds, such as carbon heteroatom bonds, but we are also interested in a formation of various or several CC bond or C heteroatom bonds. This is just one example, recent example from the area of NHC catalysis. We have developed a synthesis of highly functionalized cyclohexenones based on NHC catalysis. And for the activation using NHC, we used substrates, alpha halogenated enols that upon activation with NHC carbene generated from the corresponding triazolinium salts were able to attack in gamma position electron deficient carbon double bonds. In this case, we have used electron deficient carbon double bonds that are in close proximity to nitro-isoxazole moiety that makes this electron deficient bond enough electron deficient. Nevertheless, most methods based on NHC catalyzed approaches works in Breslow goes through the Breslow intermediate. In our case, we were interested in an extension of a Breslow intermediate to form azolium dienolates. Nowadays, we are extending this chemistry for the preparation of spirocyclic compounds, but I am not going to talk about that in details. Instead of that, I would like to show the reaction mechanism of this transformation, just to simplify the reaction mechanism. And in this case, the crucial point is here, the formation of azolium dienolate without use any oxidative method. So first, NHC catalyst generated from triazolinium salt attacks aldehyde moiety, forms classical type of Breslow intermediate. After elimination of bromine anion and deprotonation, we generate azolium dienolate. So we have so-called extended Breslow intermediate that is enough nucleophilic to attack electron deficient carbon, and then after ring closure, we release NHC carbene back to the catalytic cycle. But that's not the main area of our research. The main area of our research is the enantioselective synthesis of various types of heterocycles. I have already talked about 4-membered rings, but especially we are interested in the synthesis of 3 to 7-membered rings using either organocatalysis, including aminocatalysis, Brensted acid catalysis, bifunctional catalysis, and NHC catalysis. But also we try to combine organocatalysis with other catalytic approaches, such as transition metal catalysis. Essential part of our development of our research is definitely the application of the developed methods for the preparation of biologically active compounds or naturally occurring molecules. There are just few examples. And today I would like to focus on 2 projects. First project is focused on Brensted acid catalysis and the preparation of enantiomercury pure dihydrobenzoxazines. If we take a look to benzoxazine moiety, we can find this moiety among number of nature products and also among number of pharmaceuticals. Nevertheless, the synthesis of such compounds, it's not so straightforward. Definitely, if we take a look to traditional synthetic approaches, we can find hydrogenation methods for production of simple or simple molecules of this type. But also we can find modern methods, including palladium catalyzed aminohydroxylation or copper catalyzed cycloaddition reactions based on formation reactive species such as p-allylic systems or alanic type of copper intermediates. Nevertheless, as I said, for example, if we take a look to all of those methods, we can see that in alpha position, in position 2 next to the amine moiety, it's not possible through all of those methods to generate quaternary chiral center. And that's why we started to be interested in the use of desymmetrization of oxetanes as valuable compounds in pharma industry for the production of those dihydrobenzoxazine scaffold. For the activation of oxetane motif, we have used Brensted acid catalysis that should be able to protonate that system to make it more nucleophilic. Just note about the make it more electrophilic and to be opened by the nucleophile. Just if we compare the electrophilicity or the activation of oxetanes, for example, with oxiranes, so oxetanes are definitely more stable than oxiranes. So that's why the Brensted acid catalysis is the best way how to activate those systems. If you believe that those compounds, the starting materials, are not readily available, it's exactly opposite. The starting material corresponding oxetanone is readily available. It's pretty cheap. It's readily available from dihydroxyacetone, and through addition, nucleophilic aromatic substitution followed by reduction and reductive amination, we can easily generate number of starting materials ready to be used in this Brensted catalyzed process. After optimizing of reaction conditions, we found that that spinol-derived phosphoric acids are the most efficient for production of enantiomercury pure benzoxazine of this type. We have tested, of course, different type of Brensted acids, including bienol-derived phosphoric acid and also chiral cyclopentadiene scaffold, including Lambert type of catalyst, the catalyst where the R substituents are menthol units. But as I said, the most efficient in terms of enantio control were spinol-derived phosphoric acids. So with the conditions in our hand, we have tested the substrate scope, and I can say that the substrate scope is broad, so we can prepare different type of benzoxazines. I'm not going into the detail, but you can see that benzoxazines bearing benzyl moiety, either electron rich or electron poor, aliphatic parts of different lengths, and also, for example, strongly electron withdrawing groups such as tosyl group can be present, and corresponding amide moiety in starting oxetane is enough nucleophilic to open oxetane motif, so we can reach the corresponding product in good yield. Nevertheless, as you can see, we reduced enantiomer control of the process when we use less nucleophilic amine systems. What is also advantage of this approach that we can use this approach for production of benzoxazines bearing quaternary center next to the oxygen, and for that we had to use a little bit less sterically hindered bienol-derived phosphoric acids of this type. Nevertheless, the enantiomeric purity of the prepared compound was still good, even not as perfect as in previous cases. The application of that method could be demonstrated on for one example. Of course, we have demonstrated on several examples, but this is just to show that the developed method can be used in a bulk production in gram scale. And for example, the key intermediate for the formation of orally active prostaglandin D2 receptor antagonist has been reached in one reaction step instead of several steps involving asymmetric amino hydroxylation, ozonolysis, and then playing with protecting groups. Later on, we have also used this approach for the formation of PAM, a positive allosteric modulator of the M1 subtype of muscarinic acetylcholine receptor that is nowadays studied for improving cognitive function of patients suffering from Alzheimer's diseases. Also, based on this approach, those type of compounds can be produced. Nowadays, we are also working on extension of this method towards 7-membered rings, towards formation of corresponding diazepines or oxazepines, and few examples you can see here. So we can really play with this area a lot, and we hope that in near future we are going to present the extension to the 7-membered rings. The second part I would like to discuss here is the formation of carbocycles. And in this case, I will focus not only on organocatalytic approaches, but also I would like to shortly comment the approaches based on a combination of organocatalysis and transition metal catalysis. So why spirocycles? Not only because those compounds are common in nature. So for example, those compounds, histrionic toxins, are present in the skin of those nicely colored frogs known as the poison dart frogs. But also those compounds can be found in the area of biologically active compounds, and especially nowadays when the pharma market is moving from the flat area to the area of spirocyclic systems. Definitely spiral systems are highly has a potential in this area. So we can find several areas of application of spirocyclic systems, definitely in the area of biologically active compounds, but also in fragrance chemistry. But at the beginning, roughly 10 years ago, we were not interested in the development of new biologically active compounds, but we just started to work with spirocyclists by the accident. When we studied the enantioselective Michael additions, where we tested several types of nucleophiles to be used in phenols activated through the immunium activation, we found that under proper conditions, we are able to generate spirocyclic systems of this type, spirocyclic systems containing cyclohexene carbaldehyde moiety. So for example, when we use benzothiophenone together with 2 equivalents of alpha beta unsaturated aldehyde in presence of secondary amine and Brensted acid, dinitrobenzoic acid, we were able to generate those spirocyclic systems containing benzothiophenone and cyclohexene carbaldehyde moiety with good efficiency, not only in case of free yield, but also in high diaster control and enantiomer control. We were also interested in biological activity, well, we just tested those compounds or compounds derived from those corresponding acids, and we found that those compounds are quite active again several types of bacteria, and this antibacterial activity was quite interesting. So that's why we further studied those systems. And we focus on development of new methods of the synthesis of spirocyclic systems. So this is just the, I would say, proposed mechanism for the formation of spirocyclic scaffold through the double iminium activation and amine activation. First, activation of the aldehyde through the secondary amine gives corresponding iminium. That iminium is enough electrophilic to be attacked with nucleophilic carbon present in alpha position of phenyl benzothiophenone moiety. After formation of the first microadduct, this microadduct is enough nucleophilic to attack another molecule of activated aldehyde in a form of iminium, again to the beta position, forming this microadduct. And after either enamine formation of hydrolysis and aldol condensation, we reach the cyclohexene carbaldehyde moiety. So we can see that in one reaction sequence, we can form 3 stereocenters, and only one stereoisomer is formed with high efficiency. We have extended that system. Of course, we are not working alone in this area. I have to also appreciate the work of Professor Paolo Melchiorre, Professor Rios, Wei Wang, and many others that are also using organocatalysis for production of those systems. And recently, we have extended that system also to iminium enamine activation using haloalkyl heteroaromatic systems based on oxindole moiety. So there you can see that again, another set of spirocyclic system was formed. So in this case, 5-membered rings were formed next to the oxindole moiety. Again, the catalytic cycle goes through the iminium activation, enamine activation, and after cyclization, we got enantiomerically pure spirocycles. But as I said, or as you saw from those examples, you can see that we are a little bit limited in terms of substitution of 5-membered ring or 6-membered ring in the previous case. So that's why we started to be interested in the extension of this methodology using organocatalysis for the synthesis of spirocycles to so-called cooperative or synergistic catalysis. So in this case, we combine organocatalysis together with transition method catalysis, and we are focused nowadays on the application of palladium catalysis together with aminocatalysis, iridium catalysis together with NAC catalysis, and several other approaches. Also, copper catalyzed approaches are studied in our lab. To tell the truth, we were inspired by the work of Armando Cordova, who was interested in a combination of organocatalysis with palladium catalysis, and recently the work of Professor Jürgensen and Professor Rios who actually published the work for the preparation of highly functionalized cyclopentanes. Based on that, we come up with the idea that also diasteromeric racemic forms of those simple heteroaromatic vinyl cyclopropanes could be used as starting materials for production of optically pure enantiomerically enriched spirocycles. And we have tested various types of vinyl cyclopropanes containing heterocyclic motif. First, we started to be interested in azlactone moiety. Then we were also interested in double nitrogen containing imidazolidinones and several others. The preparation of those systems is pretty straightforward. Just through the double alkylation, SN2 prime alkylation, we can easily reach the starting materials as a mixture of diester isomers in racemic form. Nevertheless, after proper activation using palladium 0 catalyst that is coordinating with double bond system, we can easily form intermediate that is enough nucleophilic to activate it enal in an iminium form. So we were able to form corresponding spirocyclic system with high efficiency. So there is a just result of selected examples. When used as lactones, vinyl cyclopropane as lactone system, we end up with corresponding as lactone spirocyclic systems with good efficiency. Nevertheless, the diasterol control was a little bit limited. Only when we used bulky substituents on arylic cinnamic type of aldehydes in ortho position, we could reach good level of diasterol control. Vice versa, in case of imidazolidinones, we could reach single diastereomers with high optical purity using this cooperative approach. And nowadays, those compounds are studied for biologic activity. If we take a look to the reaction sequence or reaction mechanism, the proposed mechanism is based on activation of vinyl cyclopropane moiety through the coordination of palladium 0 to double bond system, forming zwitterionic species, 2. And this zwitterionic species is enough nucleophily on carbon next to the amine moiety to attack electron deficient beta carbon of the activated enal in an iminium form, forming corresponding enamine number 4, and this enamine intramolecular cyclized, giving spirocyclic scaffold. And after decomplexation and hydrolysis of the iminium, we end up with the formation of corresponding spirocycle and the releasing of both catalysts to the catalytic cycle, palladium catalytic cycle and amine catalytic cycle, as is depicted here. When we started to expand this area also to other heteroaromatic systems containing pyrazolones and thiazolones, we were quite surprised when we started to prepare, or when we planned to prepare corresponding vinyl cyclopropanes. We found that instead of vinyl cyclopropanes, we obtained those seven-membered oxepines intermediates. Nevertheless, they were obtained in good yields, but there was no single precedence for the activation of those systems through the palladium 0 catalyst. Nevertheless, we have tested our cooperative concept, and we were able to activate those oxepine moieties through the palladium insertion to carbon-oxygen bond, and corresponding second partners enals were activated with secondary amines, and we obtained corresponding spirocyclic system with high efficiency, high diasterol control, and enantiomer control. Similarly, in case of thiazolos, so when we plan to prepare vinyl cyclopropanes derived from thiazolones, we end up with corresponding thiazole motifs, again containing oxepine scaffold, but also in this case, the palladium 0 can be used for the activation of the whole system. The sulfur is not a problem for the activation through palladium 0 using palladium DBA complex. And as you can see here, we have obtained corresponding spirocyclists with good efficiency, with various type of aldehydes, and also starting pyrazolones. In order to discuss proposed mechanism, I have simplified mechanism depicted on this picture because of the space. Nevertheless, also in this case, the crucial part is the formation of zwitterionic species from oxepine moiety through the insertion to carbon-oxygen bond, and this intermediate was confirmed by infrared spectroscopy in collaboration with Professor Rojtová, and again, the zwitterionic species was able to attack electron-deficient beta carbon on imine moiety, forming similar intermediate like in previous cases, enamine that is prone to cyclize and form spirocycles, of this type, and again, after release of palladium, hydrolysis, back to the catalytic cycles, we form corresponding highly functionalized spirocycles. So that's probably the last slide of my talk related to the chemistry. So at the end, I would definitely thank to all of my students, co-workers, especially to Professor Rios from University of Southampton, where we were working together in this cooperative concept, and also Professor Roitova to elucidating the reaction mechanism of the formation of spirocycles. And thank you for your kind attention.
Great. Jan, super. What a great start. Thank you very much. And again, the presentation is now open for questions. I've sent a chat message to everybody as to how they should do that. So panelists can ask questions or attendees can ask questions. I don't see any questions coming in just now. Maybe, Jan, I could ask a question on just very much the last few slides you've had. That's a very interesting activation of what at first glances is quite an unactive double bond. So is there literature precedent for that sort of ring opening to give you those sort of oxy-palladium allyls? I've not seen them. Have you been able to exploit that for other ring systems? Does it only happen for a seven-membered ring? Would it happen for a six-membered ring, etc.?
Well, in general, there is no precedent for the opening of those seven-membered rings, and I would say that in this case, it's because of the surroundings. So because of the pyrazole motif that is prone to be aromatic. So the aromaticity definitely takes place in this palladium insertion to carbon-oxygen bond. But we have tested also nitrogen-containing scaffolds. Unfortunately, in this case, we are not able to activate, and probably there is also another problem associated with palladium, that of course, as soon as you have other coordinating atoms, including nitrogen and sulfur, you can poison the palladium, and you can make strong coordination bond between the heteroatom and palladium. So we are right now a little bit limited with the seven-membered rings used for the palladium activation in this cooperative concept.
But it's still very interesting. Well done. Very nice. Again,
thank you.
Panelists can also help by asking questions. No pressure. I don't see anyone raising hands. We have 108 attendees, just to let you know in terms of numbers. That's really good.
So probably there's something in chat.
Oh, now we have a question from Basha. In the carbocycle, what about the oxidation state of palladium?
In a carbocycle, which you mean the zwitterionic intermediate or carbocycle?
My guess is that that's what the question relates to.
So probably in this case should be palladium 2.
Yeah.
So what we assume is palladium 2, and of course, well, yeah, at least based on the opening of this opening of oxapimoid and forming zwitterionic species, should be palladium 2.
Yeah. So the Q&A function works. That's for sure. Anyone else wish to ask any questions? Yeah, for sure. Organocatalysis is a busy area. You've got a lot of competition out there, Jan. So doesn't appear to be any other questions. So perhaps again, thanks again, Jan. If I could ask you to unshare, and I'll pass over to Vladimir.
Okay, thank you very much.
Right, thanks, Jan. And our second speaker of the day is Hanna Macičková-Cahová from Institute of Organic Chemistry and Biochemistry in Prague, and she will tell us something about RNA caps. So please, Hanna.
Hello, everybody. Can you see it? It's okay. Yep. Okay, perfect. Thank you.
Yeah.
So first of all, thank you for giving me opportunity to present here our research. And I will be today actually outsider, because even though I am organic chemist, I am not using organic chemistry for the synthesis. But as already PhD student, I realized I love to use organic chemistry for understanding what's happening in nature. And what we are actually studying are non-canonical RNA caps, and we are using chemistry to understand what they are doing in cells, what is their role, and so on. So I will start with the structure of the RNA. Probably all of you know it, but RNA can be heavily modified. There are actually nowadays 170 RNA modifications naturally present in RNA detected. Among them, there are methylation and lately also reported phosphorylation of a 2' position of sugar. Then bases can be heavily modified. There could be methyls, acetyls, pseudouridines, and so on. But also 5' end of the RNA can be modified. And this is the part of the RNA modifications which is the least explored, and which I would like to actually explain you today. So here are the classical canonical caps. What does it mean, canonical? It means something natural, something regular, something what is very well described, because these caps are known from 70s. They were detected in a mRNA of eukaryotes, so it means higher organisms. What you can see here is like just imagine this is the RNA, this is the 5' end, and there is in a 5' order, not a 3' order, attach another nucleotide after triphosphate here. And it's usually G, which is methylated. This is skip 0. If you have methylation on the first coding nucleotide, this is skip 1. And if it's even on a second coding nucleotide, it's skip 2. These caps are essential in even our organisms for regulation of nuclear export, for prevention of degradation by exonucleases, and they are also recognized by translation factors, and they are triggering the translation. And I think their importance is nowadays quite known, because they were also essential for the development of vaccines against COVID and in future against many other diseases. Cap 0 is somehow unfinished Cap 1, because it doesn't have the methyl, and our bodies are having so-called innate immune system, which is recognizing it and saying to our body, hey, there is invasion of the virus. This is not the real cap how it should be. So you can see that even tiny changes can play an important role in what these caps are doing within the cell. Till 2009, it was believed that there are no caps in bacteria and that the caps I mentioned are the only known. But in 2009, one group of Professor Liu from Harvard found that actually RNA from bacteria contains NAD, nicotine-amide adenine dinucleotide, and coenzyme A. How they find it? By mass spectrometry. So they isolated RNA from 2 types of organisms. They digested, they chopped it by nuclease P1 into the particle building blocks, and then they put it on MS, and they saw it. I have to say that in the time I met many biologists and biochemists, and they were claiming, this is nonsense. We really think that it was just contamination, because these are actually protein cofactors, which are working with proteins, with amino acid proteins. But I said, no, look at it. As a chemist, you notice that these are actually RNA molecules, both NAD and coenzyme A. They both contain RNA in themselves. So probably first they were part of the RNA, and then only lately they became the protein cofactors. Okay, so for me, it was super interesting. And I started my postdoc in Heidelberg, and my task was to kind of understand these molecules, because we didn't know anything about the sequence of the RNA, which are having NAD coenzyme A. We didn't know the structure of these RNA. We didn't know biosynthesis, or how these molecules are getting there, or how they are biodegraded. So I approached it as a chemist. So we needed specific technique to capture only NAD RNA, something what will selectively attach something very special to NAD molecule and not anywhere else. For example, triple bond, because you cannot find triple bond anywhere else in organisms. So if we manage to attach triple bond to NAD, Then we can do classical Kappa-Click catalyzed reaction with the biotin azide. We will attach biotin to the RNA, which had previously NAD. And then, because biotin is making very strong bond to streptavidin, so you can have kind of streptavidin beads, which will bind to biotin or biotinylated RNA. You capture it and you wash out everything what doesn't have the biotin. I spent almost 1 year with this method. The method, because must be very selective, so it must work only with NAD, and sensitive, because RNA is very fragile, and even if you touch RNA by your bare hand, you will destroy it, because RNases are simply everywhere. And it was also necessary to have positive and negative controls. Finally, I was successful. I found the enzyme, which is called ADPRC, adenosine diphosphate ribosyl cyclase. And this enzyme was known to hydrolyze NAD. But I forced this enzyme to do transglycosylation reaction. So instead of nicotinamide, to attach their propargyl alcohol. So nicotinamide is the leaving group. So I was very happy that this was working. And because it's enzyme, it was very selective. It didn't work with A, it didn't work with C, G, and so on with other building blocks of RNA. And then I was testing if the propargyl alcohol is really the best one. So I took the whole portfolio of these alcohols, and I actually found out that the highest yield I'm able to get if I use the pentanol. And then I performed Kappa-Click catalyzed reaction. This was working perfectly. I did it on a NAD, then I did it on NAD RNA. Everything was working. So this was the moment I was able to go to the RNA from E. coli. So I isolated RNA from E. coli, and I applied on it ADP ribosyl cyclase and pentanol. So I attached to the NAD RNA triple bond. I performed Kappa-Click catalyzed reaction. I captured this RNA on the streptavidin beads, that all the other RNAs which did not have NAD, I washed out. And then I performed the combination of molecular biological reactions, which led to the preparation of so-called a library. So it's a library of RNA or DNA sequences which you sequence on certain platform, in this case on Illumina platform. And we did it, and what we have found is actually that the NAD is attached to small RNAs, which are having rather regulatory role, and also on some small RNAs which are coded on plasmids, and we also detected some short fragments of mRNAs. Okay, this was nice, but we had a question. What is the role of the NAD there? And the first idea which came to our mind was that probably it plays the role of this cap, as this canonical cap I explained before in eukaryotes. This is doing the same in bacteria. So it means it's protecting the RNA from the degradation. So we first tested the enzyme which is responsible for the degradation of the RNA in bacteria, RNase E, and we indeed found out that NAD RNA is much more stable than triphosphate or monophosphate RNA. Then we tested RPPH, enzyme from the Nordics family. I will get to these enzymes. They are super interesting. And this enzyme was known to cleave triphosphate RNA into monophosphate. So can this enzyme cleave NAD RNA? We tested it and we found out that actually it can't. Okay, but if there is NAD RNA, there must be an enzyme within the cell which will be cleaving it. And we came up with an idea that there is, from this Nordics family, another enzyme known, which is called NUC, and known to cleave free NAD within the cell. So we came up with an idea, what if this enzyme is not actually cleaving the small NAD, but it's cleaving NAD, which is part of the RNA. So we tested it, and indeed we found out that the NAD RNA is a substrate of NUC. So we came to the conclusion, it was year 2013-14, that actually E. coli is playing with its RNA as like higher organisms are, and it actually can decide what information to keep. That it's not like when there is enzyme for the degradation, all the RNA would be degraded. It can really choose what will be degraded. So if the information in RNA is encoded in RNA with a 5' monophosphate, then such RNA is immediately degraded. So cell is losing the information which it does not need. If the information is in RNA with a triphosphate, then there must be first RPPH expressed. So the cell must decide, okay, I need to get rid of it. I need to make RPPH, it will cleave it, then it will be degraded, again by RNase E. But if the information is in NAD RNA encoded, then there must be even other enzyme. It's NUC. So the cell says, okay, I need NUC. It prepares NUC, express it, cleaves NAD RNA, which will create monophosphate RNA, which will be degraded. So it has like a potential to decide what is important, what is not important, what should be degraded, what should stay. So this is like we managed to publish it by the beginning of 2015. And this method has been actually applied almost to all organisms nowadays. So we know that human cells are having NAD RNA. We know that plant cells are having NAD, and we also know plethora of bacterial cells who are having it as well. And also archaea are having NAD RNA. So what we are, I think, now missing are some kind of parasites. So it's still waiting the field for that. But in general, NAD RNA is simply everywhere. We know that it's probably RNA polymerase, which is incorporating it into RNA. We know many biodegradation enzymes like NUC and many other, but we still do not understand its function. I will not go into the detail with this because we also did some work about it, and we are, I would say, quite close to understanding its role, especially in higher organisms. But I will be talking about something else, what we did with our team. Because when I established my team in 2016, with my two postdocs, we noticed very interesting thing. Then in that time, these 4 known RNA caps are all cleaved by Nudix enzymes. And we said, okay, these are very interesting enzymes. What these enzymes are doing? And we found out that they are in all classes of organisms. They are very often called nucleoside diphosphate linked to another moiety, hydrolases. E. coli has 13. We have 24 of Nudix enzymes. Some of them are related to cancer, but we don't really understand the function of all of them. And when the first was described or discovered, it was believed that they have house cleaning function, but it doesn't look like that. We have 24 house cleaning Nudix enzymes. They can cleave wide range of organic pyrophosphates, including penta-inositol phosphate, coenzyme A, FAD. Some are decoupling enzymes, but they can also cleave dinucleoside polyphosphates. And these molecules attracted our attention because if you again look at them as a chemist, you realize they are having strikingly similar structure to what I described at the beginning, 2 classical canonical caps, which are known from 70s in all eukaryotic cells. They have again the ribose, which is attached by 5' phosphate bridge, and there is usually adenine and guanine, so usually purines. There's not much known about pyrimidines. And these molecules are known for more than 50 years. They were detected in human cells, they were detected in plant cells, they were detected in bacteria, simply everywhere. But we still do not understand what they are doing. We know that they are biosynthesized by tRNA synthetases or by other processes. For example, ubiquitinylation can also lead to their formation. They are cleaved by this Nudix enzyme. Very often they are called alarmons because they are somehow connected with the stress. So when the cell is stressed, then their concentration increases. But we still do not understand how this is working, because just imagine when you woke up this morning, there was an alarm ringing. There was your ear who was recognizing the alarm and waking you up. It took the action actually. So there is the alarm, and we do not know what is the ear in the cell. So this is still unknown. So we decided to look at them from the RNA point of view. Yeah, and then, moreover, we found out that in literature is described that they are present in very high concentration. So we said, okay, if they are present in a high concentration within the cell, can they be simply taken by RNA polymerase and for the building of the RNA as a first non-canonical initiating nucleotides? So we tested various RNA polymerases, and we found out indeed they are very good substrates, and such a RNA is very easily formed. So okay, so the answer to the first question is okay, RNA polymerases can take them, incorporate them into the RNA as the first non-canonical initiating nucleotides. Okay, are there Nudixes which can cleave them? So we went to this table published in 2006, when there was shown that there is one particle, these are all Nudixes from E. coli, and one of them, NUT-H, is known was known to cleave 3AP5A, AP6A, AP4A. So something we were interested. And we noticed that the transcript abundance is pretty high. So it's a very abundant enzymes. So I said to my students, okay, we have to purify this enzyme and we have to test it on RNA level. My students came and said, no, we do not have to buy it. We have it in the freezer. Actually, nowadays, its name is RPPH. Maybe you remember I mentioned it, that it's the main enzyme which is cleaving triphosphate in bacteria. And this was like the first report in 2008 that it's cleaving triphosphate. Later on, the same group said, oh, it's not triphosphate, it's a diphosphate, what is cleaving mainly in bacterial cell. But as you see, like, in the field, it's not so clear. We still do not know how the RNA looks like in bacteria. So we took this enzyme, we tested on a capped RNA, and we found out that it's cleaving excellently all the capped RNAs, and is actually preferring those caps which are having between 4 and 6 phosphates. Then we knew about another enzyme, which is called APAH. This enzyme was known to have some, again, connection to the stress, because if it was mutated in bacteria, there were general defects in the stress resistance and the cells were dying from it. And we know that APAH is cleaving 3AP4A. So we were testing whether it will be cleaving dinucleoside polyphosphate capped RNAs. And we tested, and indeed, we found out that it's again cleaving all the capped RNA. So we know these small molecules exist in a cell. We know RNA polymerase can incorporate them into the RNA. We know that there are enzymes which can cleave it from the RNA. So there is basic question remaining. Are they exist in vivo? Can we detect them? So for these purposes, we decided to go, of course, to the simplest model organisms, to E. coli, to bacteria, and we grow bacteria. In an exponential phase, the bacteria is super happy. It has everything. Exponential because it's exponentially growing. There is enough space, there are enough nutrition, it's just perfect, it's a dream, it's a heaven for bacteria. But at a certain moment, the bacteria gets into the state, and there are too many. There are too many fellows around, and they start to dying. And they are like spreading, all the toxins and so on. The nutrients are already consumed. This is the stationary phase. And if the curve continue, it would go down and you will see the phase of dying. But the stationary phase is actually a phase which is more or less, I would say, the most similar to what we see around us, because nothing is ideal. And it's also somehow simulating the stress. So we decided to isolate RNA from the exponential phase. So it means ideal phase, heaven phase, and a stationary phase, late stationary phase. We isolated RNA and we divided into fractional short and long RNA. So the threshold is around 200 nucleotides. And we then focus on short RNA. Why? Because just imagine what we are doing again, I will get to it, but we are actually taking RNA, we are chopping it into the nuclear building blocks, and then we are putting the whole mixture into MS, and we are trying to find the caps. But if you have short RNA, there are 200 nucleotides maximum and 1 cap, so 1 per 200. If you have long RNA, these are thousands, 2,000, 3,000 of nucleotides per 1 cap. What does it mean? You have much higher noise, much higher background, and it's much more difficult to detect anything, and it's super difficult to detect these molecules. So that's why we decided to focus on short RNA. We discarded all small molecules. So we try to really wash it perfectly, because whatever we will see must come from the covalent bond to the RNA. Then we divided the samples into 2. One we treated with nucleus P1, one we didn't. And if we would see something in a sample where we did not put nucleus P1, it means that it was not washed properly, and we have to do it. We have to add, for example, urea to wash it more, and so on. And then we perform comparative LCMS analysis. And indeed, we found out 9 new types of RNA caps. We call it a new class of RNA caps. Some of them, like AP3A, AP3G, and AP5A, were in that form. But there were other 6 which were containing another methyl or dimethyl, which we didn't know where it is. So what we did, we either prepared chemically the standards, because we were presuming what could be potentially positions where the methylation could be. We took inspiration from what was already known in literature. Some standards we had to buy because we were not able to prepare them. It was like a custom synthesis. And then based on retention time and fragmentation of standards in LCMS, we were able to conclude that dimethylated GP4G is actually M7-GP4D methoxy, and methylated AP3A is M6AP3A. So just by naked eye, it was visible that there are more of these caps in a late stationary phase. So it means in a stress phase, and not only more types, but also like by the quantity. This is expressed here when we compared the amount of the dimethylated GP4G in RNA from exponential and late stationary phase. In both cases, we are injecting 500 micrograms of RNA, and in then you can see in a late stationary phase, there is twice more of this cap. Just by, and then we were wondering what is the role of these caps. And I think this is the question for couple of years. I will show you how we are approaching it, but it's not so easy to answer this question so fast. But we at least thought that maybe we can understand what the methylations are doing there, because we know some hydrolysis, this RPPH, IPAH, which can cleave dinucleoside polyphosphate RNA. And we had to question, okay, they can cleave it if they are not methylated. What will happen if these caps are methylated? Will these enzymes be stable, able to cleave it? So what we did, we took RNA from E. coli and we spiked their GP4G RNA as a model RNA, and we added these enzymes, RPPH and APAH. And what you can see that GP4G was perfectly cleaved by APAH and partially by RPPH, or like from majority 75% after 1 hour. But M7-GP4D methoxy was not cleaved at all by RPPH and cleaved by APAH. We had a look to expression profile of these particle enzymes in the growth stages of bacteria. And we were able to build some kind of story or hypothesis for the role of these methylated caps. So what we think is like that in exponential phase, in this heaven phase where everything is perfect, bacteria simply prepares RNA with various caps. When it does not need it, RPPH cleaves it, nucleases degrade it. Perfect. You know, it doesn't need the information, doesn't need the RNA, it's degraded. It's like a really flexible reaction to the environment. But when the situation changes, it means that it will get to some starving phase. It will not have enough building blocks and nutritions. Some unknown methyltransferases are activated. They methylate this GP4G. RPPH cannot cleave it. Nucleases cannot degrade it. Bacteria doesn't have the RNA of which it would need, but the problem is it doesn't have even the building blocks for new RNA. So at least it keeps what it has. So it cannot react flexibly, but at least it has some RNA, it's protected. But when it goes back to normal condition, to like not normal, this ideal condition with enough of nutrition, in that moment, APAH is expressed and produced in bacteria, and then these methylated caps are cleaved and nucleases can degrade it, and the cell is back on top. It means that it can flexibly react to what happened or what's happening around. Okay, so this was our finding, and with this finding, we started to have many questions. What is the role of these dinucleoside polyphosphate caps in bacteria and also in mammalian cells? How these caps are made, modified, and metabolized, how these metals are getting there, and what are the connections between them and stress response. And for this, we need chemistry, because we need techniques which will help us to identify the sequence of these RNA which are having that. Are these tRNAs, mRNAs, SNORNAs, and so on. So I will show you now how we are approaching it. It's still not published, it's still not done or finished, but I believe that we are on good tracks. So what we can do, what we can use for it. We can use organic chemistry, we can use enzymes, but just imagine you are looking at AP4A RNA. You have their AA, you have their ribose, you have their phosphates. It's everywhere. It's in whole RNA. It's simple, and the basic building blocks are like this. So what type of chemistry you want to do? it's difficult. Enzymes, maybe. Then you have to have very selective enzymes. You have to explore like a big portfolio of enzymes to find something that will be really selective one for one particle cap. But what is typical for the cap is this vicinal dial, because if you look at the complete RNA, there is always this 2' OH, but no vicinal, because the 3' is actually bound within the phosphate backbone. But here, the vicinal dial is free, and this is allowing us to use organic chemistry. Because what we can do, we can selectively oxidize it. If you have a look at the RNA, you realize there is another vicinal dial. It's at the 3' end. So first, what we have to do, we have to blind it. So we are actually adding there 2' deoxy A, which doesn't have vicinal dial. So in that moment, indeed, now vicinal dial is just in the cap. And then we perform oxidation, selective oxidation of vicinal dial, bar periodate, and then we do reductive amination with the oligonucleotide, which is having amino, or now we know that hydrazide is better. And then we can capture the RNAs which are having this oligonucleotide attached, wash out everything else, again on the streptavidin, like a complementary probe, because now we know the sequence. And then we can prepare the samples for nanopore sequencing, which is very special because it's direct sequencing. So these are like 2 pores, and through it is like a DNA or RNA going. And now we know that they will be different signals if they are diphosphates, if they are triphosphates, if they are tetraphosphates. There is different signal if the cap is AA or is GA or it's methyl GA. So we will be able, we need to use machine learning and so on, and prepare like a new kind of software, but we will be able to know what are the signals of particle caps, and then we can take the cellular RNA and say, okay, this particle RNA is having, for example, NAD from 5%, from 10% is AP4A, and the rest is, for example, canonical cap. So this is one approach. Second approach is kind of enzymatic. It's very similar to what we did with or not very similar, it's partially similar to what I did with ADP-RC. So what we have RNA. And first of all, we can cleave all the phosphates which are at the 5' and all the caps. And then we can take, for example, one particle enzymes, it's 18-NUT6, so again, Nudix enzymes, which we know it's selectively cleaving only AP4A and nothing else. And if we have this enzyme, and we found it already, this enzyme is doing it. So if we use it, then we will have phosphate, 5' phosphate, only at the originally AP4A RNA. And then we can use the ligation reaction to it, and then we can sequence it. So this is another approach we are taking. And the last one is actually using so-called fingerprint, because RNA can be read by reverse transcriptases, and they are making from the RNA cDNA. And we already know that there are reverse transcriptases which are reading our caps differently. So for example, Enduro is reading AP4A like as you can see here, this column on the left side, and Protoscript is reading completely different. So if we do the combination of these reverse transcriptases, we will get the fingerprint, and we will know that one particle RNA is having, again, AP4A, M7GP3A, NAD, and so on. And again, we will use nanopores. And then we can take the isolated RNA. So this is my last slide. No, there is one more, sorry. Conclusion. What should be the take-home message? That the RNA in all organisms heavily modified at the 5' end. That is really depending on the environment, what's happening around the cell. That the general role of these caps is so far unexplored in bacteria and in eukaryotes, and we really need chemistry, we need techniques for identification of these capped RNAs, and then we can further explore what they are doing, what is their role. So with this, I would like to thank mainly my team. I have really a team of wonderful people, former members of my team, grant agencies, our collaborators, and again, you for inviting me here and for your kind attention. And I will be happy to answer any question.
Super, Hannah. Very nice presentation. Excellent examples of chemistry solving biological problems. Again, we're open for questions and answers. So we have some previous ones relating to Jan's presentation. So no further ones there.
It's too biological.
No, well, look at again, none coming through in the chat, and nobody's raising their hand. So well, I mean, I'll again start by asking a question. So methylation is it's very important in like SAM and all the acetylmethionine, all those sort of things. Why do you think nature has developed these differently methylated? Is it some sort of code, do you think?
Yeah, could be code. They could be recognized by different enzymes. Just imagine there is some decoration. This is what really is happening. Nowadays, we know it in mRNA. There is M6A, and there are really readers which are binding it. And then they are preventing for the translation. So simply they bind and they block it, for example, or they activate it, or they take it somewhere else. So it's really, it's a kind of additional code. We call it additional layer of information on RNA. That in RNA is not only the information which is written in the sequence or the secondary structure, but it's also written in these modifications which are around.
Very good. Oh, questions come through from Basha. Again, very good, Basha. Enzymatic cleavage of phosphate group is selective or is it specific?
Okay, maybe I'm not using the right expression. It's specific in a way, it's cleaving only AP4A or selective. I actually probably I don't understand what the difference between selective and specific and how does he mean it?
Yeah, I'm not sure either. I just read the question.
Oh, yeah. So it's cleaving just AP4A. It's not cleaving AP3A. It's not cleaving any other cap.
Right.
Like that.
It's a very selective and specific answer.
Thank you.
Very good.
Maybe if I may just congrats Hannah on really exciting and complicated story. If I take you back in time, the athlesia enzymes that help you to put in the propargyl group, you mentioned that it's sort of nucleotide specific, selective, differential. So is there any explanation for how the enzymes picks up this type of selectivity?
Probably this enzyme is the selectivity comes probably from the positive charge on nicotinamide, and as the others do not have it, they are not binding there. So probably there will be some amino acids will be which will be negatively charged, will be interacting with it. So for example, it's not working with NADH, which is not positively charged. So we know it's not taking it. So probably this is the key within the active side of the enzyme.
It's a cool story. I like it.
Thank you very much.
Great, thanks, Jakob. And every Friday morning, by the way, we have our group meetings. So my body's very used to getting a coffee at this hour of the morning. So thanks to my two PhD students, Neve and Aveen, who just dropped in a coffee. Thank you very much. Okay, just keeping to time, we might stop there, Hannah, sorry if that's okay. And thank you again for excellent presentation.
Thank you.
Over to you, Vladimir.
Yeah. Okay, now is the time for my colleague, Jakub Švenda, from Masaryk University Faculty of Sciences, and Jakub will take us to the world of natural products. So please, Jakub.
Right, I just want to make sure that the slides are clearly visible.
Yes.
Okay, thank you. And the laser pointer on. Yeah, so thank you very much for allowing me to join this community of Czech chemists and biochemists and biologists. So I'm interested in natural products, and before I jump into it, I wanted to mention that there is a lot of other chemistries happening in the Czech Republic and also at the Masaryk University. The organic chemistry section has some very, so that's my home department, has some very solid and fine medicinal chemistry that is led by Kamil Paruch. We have a long-term expertise in organic photochemistry. That is the lab of Peter Klan. Vladimir, who kicked off the morning session, is excited about supramolecular chemistry. We also have synthetic material chemistry done by Tibor Mazal, and my lab is primarily interested in natural products chemistry and biology as well. So I thought I would start by telling you why we are interested in natural products in general, and this will be probably not surprising to many of you. Natural products remain an important source of bioactive small molecules that find many applications as research tools and also in human medicine. And this biological activity can be tied to, or often is tied to, a specific structure of the small molecule natural product. And this can be a very simple molecule or a fairly complex one, as is the case for strychnine I'm showing here. And the way the molecule is shaped, the way it looks, doesn't really change with time. And that is referred to as molecular complexity, and it's an intrinsic property. Now, if one decides to prepare such molecules in lab, which you may want to do for different reasons, either reaching the natural product itself or modifying its structure and therefore properties, one has to deal with what is called the synthetic complexity. That is, how difficult is it to assemble the natural product itself from commercially available materials. And the good news is that this can change with time, and I think we are getting better at it. As I'm highlighting here, with sufficient effort and advances in strategies and methods, you can streamline and simplify the synthesis of even fairly complex molecules. And I've picked two of the many wonderful syntheses of the strychnine itself. And since these are highlights in chemistry, I'm going to highlight 3 projects that we have worked on over the years without going in too much detail, but hopefully this will give you a sense what we are interested in. So the first story I wish to tell you about has to do with a family of natural products called pseudotins. They are fungal metabolites, and I'm showing a few of these natural products here. They are interesting because they display all kinds of biological effects, primarily in cell cultures. And this was one of the reasons people have been interested in investigating these further. It should be noted, however, that for most of these effects, the underpinning mechanistic explanation, how it actually happens, is poorly understood, and it remains to this day. So we thought that if we could have chemistry that would allow us to prepare these types of structures, either in natural form or modified, labeled, and so on, this could be useful in future chemical biology studies, for example. So thinking about the synthesis of these molecules, you will notice that they carry different names, but they have a lot in common. They share this common center spirocyclic core, where you have a furanone ring attached to a gamma-lactam. And then they differ in terms of stereochemistry, and they differ in terms of the specific substitution present. And there were many syntheses of these molecules published already. And I'm not going to have time to tell you about all of our work in the context of serotins, but I thought I would focus on this spirocarbon that links the 2 rings together. That's a quaternary stereocenter. Anyone who makes serotins has to go at some point through it and find a way to construct it stereoselectively. So Petra and David in my lab found a way to do it. They utilize these rather reactive and unstable electron-rich furans that are highly nucleophilic at this position. When combined with alkynones, they undergo a 1,4 addition, the Michael reaction essentially, and yield the corresponding products that already contain this quaternary stereocenter, or if you like, the spirocarbon 2b. And you can catalyze this reaction by using nickel-diamine complexes developed by David Evans and Daniel Seidel. And by choice of the chirality within the diamine ligands, you can control the absolute configuration at the quaternary center with acceptable selectivity. And the method is has a relatively broad scope, and I'm not trying to overwhelm you here with data, but these examples show that you can have different substitution either at the furanone component or the alkynone, and the selectivity remains for the most part very high, meaning enantioselectivity. It's also interesting to note that oftentimes the method prefers the formation of kinetic Z alkenes in these conditions. And having flexibility is very important if one is trying to support biological explorations with synthetic chemistry, because all of these could be in principle elaborated to the natural and unnatural pseudotins or analogs thereof. So I'm going to show the application in the context of natural compounds due to the lack of time. So in one case, we have used this specific furan, electron-rich furan, which has actually another furan attached to it. And using the RR nickel catalyst, we've established the quaternary stereocenter. This was done by David, Jirka, and Petra. And from here, in 5 steps, we could arrive at the natural serotin cephalomycin C. What I would point out here is that this quaternary stereocenter sort of transmits the stereochemical information onto the newly formed adjacent stereocenters next to it. That's a powerful stereo control element. More recently, we got interested in an interesting problem of another natural serotin. It's called serotin A2. It was isolated in 2011. It's a little bit more complex, at least stereochemically. It has 5 different stereocenters. And people have been interested in biology of this molecule in the context of anti-seizure effects. The issue that came up is in 2016, there was another report describing the same natural product and revising not 1, but 3 of the stereocenters, their configuration. So there were 2 proposals in the literature, as you can see, where all these 3 stereocenters are now suggested to be the opposite, actually. And so obviously, if anyone is interested in exploring the biological activity of serotin A, it's important to know its structure. And this was a perfect case for the nickel chemistry, because we used properly substituted furan in the 1,4 addition, and using either the SS or RR catalyst, we would establish the spiral carbon configuration, and then that transmits the stereochemical information to the rest of the lactam ring. And this way, we have been able to support the 2016 stereochemical revision. So at this point, I thought I would turn the page, literally, and tell you about a second project that deals with a natural product for scoline. So what you're looking at here is an X-ray crystal structure of an essential enzyme called adenyl cyclase. It's part of the enzyme. It catalyzes the infamous and important biochemical reaction, ADP to cyclic AMP conversion. The enzyme has 9 different variants, isoforms. And here you can see the active site with the ATP bound to it as the substrate for the biochemical reaction. And the enzyme itself is modulated by G proteins, but also can be influenced by small molecules. And forskolin is a natural product that actually stimulates the activity of this enzyme. And not surprisingly, forskolin became a standard research tool in biology to increase levels of cyclic AMP in the cell. So with the knowledge of how forskolin functions and where it binds and how it actually interacts with the enzyme itself. And importantly, having a lot of functionality on it. It comes as no surprise that semi-synthesis was a go-to method in modulating and improving properties of forskolin itself, and hundreds of analogs can be made. Some of these are used in research. There was one derivative shown here that made it in Japan through clinical trials. And when we approached this project, we were aware of the power of semi-synthesis. We were looking where might be the limitations of the method. That is, what semi-synthesis might not be able to do, and where de novo chemical synthesis might offer something new. And so we started to think about how would one put together the molecule of forskolin in the lab from simple starting materials. And we were not the first. We had the benefit of prior contributions, landmark syntheses of many other labs. If I simplify the ideas behind the synthetic design, you can start with the left-hand ring and sort of try to build up the forskolin structure from left to right, if you will. And this was done in numerous works that have been reported. You can also start with the middle ring, the central ring, and then sequentially append the left and right hand cycles. This actually turned out to be approach we have taken recently, where the central ring comes from this dimethylated benzoquinone, and we would use a Diels-Alder reaction with a diene to append the A ring of forskolin, and we would utilize isoxazole as a masked 1,3-dicarbonyl to assemble the C ring or the right-hand cycle of forskolin. So this took us about 24 steps from commercially available materials, which at that time was the shortest synthesis. And there was some nice work coming from Pronilin Lab 2 years later, who streamlined the work further. We have at this point teamed up with lab of Kamil Bar, colleague of mine, and while Ondřej and Roman developed the synthesis of forskolin, Kamil with his students Lukáš and Vyacheslav took on the challenge of investigating fully synthetic analogs, and I'm showing two examples here. So by changing the diene structure as the most obvious way, I'm showing two examples right here, you can arrive at molecules that actually formally delete this characteristic gem dimethyl group, and you either have a CH2 group or you install new functionality there. Obviously, this is no easy walk, and it's a hard work, but it has given us access to molecules that would be hard to imagine by semi-synthesis. And I think excitingly, these two synthetic analogs still stimulate adenylate cyclase, but the isoform selectivity profile is different relative to forskolin, and that is something that really has been propelling the research on this particular project. So I don't have much time to go into greater detail in terms of biology, but I thought I would also mention more recent approach that we have taken. We have had another look at the way we put together forskolin structure, and we have proposed that if one could pre-assemble the left and right hand cycle and find a way to connect these together, bring them together to create this tricyclic scaffold of forskolin that might be useful in some context. And this is a work that Pavel, Ondřej, and Roman have contributed to. So I'm showing one example where we combine the left gamma hydroxy enol as the left cycle, and the right-hand cycle is recognizable, but it's in the form of a bicycle, and that is absolutely key for the fragment coupling to happen. So this is done via initially transesterification between this hydroxyl and the ester group over here, followed by intramolecular Michael addition. And when things are properly set up, the reaction works very well on a gram scale. And it also displays, I think, pretty useful flexibility in terms of what kind of methylation patterns and substitution you can have in the left or right-hand fragment. And I'm showing a few examples here. What is also notable is the level of stereocontrol that we sort of consistently see. So you set up 3 contiguous stereocenters. One of those is all-carbon quaternary. And in all of these examples, we see exquisite level of diastereoselectivity that can be explained after the fact. So having access to these fragment coupled products, we envisioned that it would be relatively easy after Wittig olefination of the original aldehyde to essentially isomerize this molecule into the tricycle forskolin-like scaffold that is tethered still via the lacton ring. And the missing link here, we thought, would be olefin metathesis. So imagining ring opening metathesis here would generate this triene that would now close by an alternative combination of olefins to make the tricycle. This was an ambitious proposal that initially was a trouble because actually these bicyclic 3-to-1 bicyclic systems are rather reluctant toward ring opening metathesis, to our surprise. Eventually, the students found that if you convert the ketone into the corresponding cellulose ether, you presumably pick up some sort of a ring strain, and that allows you, under moderate pressure of ethylene, to use Hoveyda-Grubb second generation catalyst to perform ring opening olefin metathesis, that is ethanolysis, to arrive at the triene that we wanted. And at this point, we would isolate the triene and expose it once again to Hoveyda-Grubb's second generation catalyst to have an unpleasant surprise of regenerating our starting material rather than forming the desired product. So there was a fundamental selectivity issue in this metathetic cascade, or in this metathetic pathway. And fortunately, the solution turned out to be very simple. This cellulose ether containing very weak CH bond over here, doubly allylic and alpha to the ether oxygen, is readily oxidized, and you get slightly different oxidized form of the substrate, which due to geometric reasons just cannot close back to the bicycle and furnishes the desired tricyclic compound. So these are 2 highlights from the synthesis. There's much more in this work, and I apologize to my students for not being able to talk more about it. But overall, we have chemistry that is obviously not trivial, but it's useful. We find where you take the cyclic starting materials and you assemble, as I showed you, the tricyclic lactone tethered core, and from here you can branch into different molecules, and some of these are really close in structure to forskolin, and so by testing their activity and potentially isoform selectivity, we are enriching the structure activity relationships that are available currently. Okay, so I'm gonna turn the page one more time to tell you about another natural product family represented here by the structure of bactobolin A. So this is also a secondary metabolite. It's an entire family called bactobolins. And here's the chemical structure. What you see here in the image is zoom in onto a bacterial ribosome. And you can see that binds the bactobolin A and engages in a lot of stabilizing interactions. And that leads overall to inhibition of translation protein synthesis. And what got us interested in bactabolin specifically was that the binding site is different from the binding sites of approved antibiotics on the market. And that would be sort of the window of opportunity to develop small molecules that target well-established molecule target in antibacterial antibiotic research. Of course, it also had issues. In probably the most significant one being poor selectivity between bacterial and eukaryotic ribosomes. And so one idea would be to change the structure of bactabolin in a way where it doesn't lose the activity, but hopefully can do something with this problem of selectivity. So it's now a little bit over 2 years that Petra essentially single-handedly converted natural product quinic acid into bactabolin A in 16 steps and 10% overall yield, which remains the most efficient route to date. And I unfortunately don't have the time to tell you about all the details of the synthetic work. I'm only going to highlight one or two steps from the synthesis that I consider interesting. One of them is performed on this advanced intermediate. It's an open chain carbamate that under the conditions of Dubois amination undergoes site selective and highly diester selective amination to make this oxazolidinone. So it inserts into position shown in red. And Petra has used this intermediate, which I redraw, which is redrawn over here, to perform ring isomerization. So these two rings, when activated using the sulfonyl group, that activates the carbamate or the oxazolidinone for an attack by an enolate. That's an intramolecular transformation inspired by work of Weinreb and Dale Ward. But Petra found a wonderful one-pot procedure how to do the activation and isomerization in one pot. And you can probably imagine how going from this synthetic intermediate to bactabolin A is relatively obvious. So this chemistry we have used since repeatedly and accessing bactabolin A and also some of the more complex members of the family. Quite recently, we had a second look at our synthesis, and we liked the end game of the synthetic route, but we are thinking whether there would be any opportunities to make shortcuts to streamline the synthesis further. And one of the intermediates that we kept looking at was this oxazolidinone that Petra prepared via amination. And we have proposed or speculated that it could be possibly made via targeting this strategic bond where you would work with something like a alpha amino radical, which tend to be highly nucleophilic, and the corresponding electron deficient olefin. So this would be set up for a Giese type reaction. We have tested this approach by preparing two fragments for this transformation. Again, starting with quinic acid, we made the corresponding electron deficient alkene. And in the right arm of the synthesis, we have taken threonine, and in 2 steps, we have made the oxazolidinone carboxylic acid. And then under the method of Dave Macmillan, we have used photoredox catalysis to perform oxidation of a carboxylate anion to carboxy radical, which then instantaneously decarboxylates and leaves the alpha amino radical over here, which then Michaels into the electron deficient double bond. And this was done by Abakar and Andre in my lab. And out of the 4 possible diastomers, we observed 2. These are readily separated. And one of these diastomers is congruent with the stereochemistry of the natural product, one of these bactobolins called actinobolin. And that has given us a streamlined route to these natural products. We now have less than 10-step synthesis and approaching the overall yield of 20%. So I think this is going to be very useful. We are using this chemistry to walk around the molecule. I'm showing just a sample of structures that we have already published on, and it's a comparison of structure versus ability to inhibit bacterial and eukaryotic ribosomes. So we use two types of bacteria and eukaryotic ribosomes. This is done actually in the lab of Sven Hobby at the University of Zurich. And so the lower the number, the more potent the compound is. And so here's one of the natural bactobolins. And you can see that these are all made synthetically. And then, for example, by incorporating 2 fluorine atoms, you can do a lot to potency and selectivity of the inhibition. And you can also learn what not to do, such as the case of the larger substituents shown over here. I think I'll be running out of time pretty soon, and I want to make sure that I leave enough time to thank the past and present group members. I think they should be proud of what they have done. I'm very grateful to my colleagues, especially Kamil Parov for long-term collaboration and support, Lukáš Kubala from the Czech Academy of Sciences for biology support within the Pserotin and forskolin projects. I'm grateful to Eric Badger and Sven Hobby for the ribosome testing, and financial agencies for the support. I want to specifically thank the Bader family and the Bader Philanthropies for believing in what we do here, and really to anybody who found time to tune in. And thank you, Pat, again for conceiving this idea of highlighting organic chemistry in Europe and running with it.
Great, thanks very much, Jakob. Excellent presentation. Always interested in natural products chemistry myself. Basha is straight in with a question. Well done, Basha. In bactobolin, what is the wavelength of the blue LED used in the reactions?
Okay. Oh yeah. So these are blue LEDs. They are not specific wavelength. So it would be a spectrum. It would be corresponding to the blue light. It's actually a lamp used in aquariums where you grow corals. So it's nothing sophisticated, and you only need to match the absorption of the iridium catalyst, and so it's a very simple setup and extremely useful.
Great, and Basha is straight in with another question, which I'm
Yeah.
not sure what's been asked. Your studies in forskolin, whether it's come under truncated analogs or it's elongated analog.
Could you read it again, Pat?
I mean, it's not that clear. I'm not sure what's been asked either. Your studies in forskolin, whether it's come under truncated analogs or it's elongated analog. I'm just reading out exactly what it says.
Right. I know it's not your question. Yeah, I don't understand it, so it's difficult for me to answer.
No, I don't either. So maybe Basha have another go at asking the question,
Right.
if you don't mind. And again, please, it's open to others to also ask questions, also panelists.
May I?
Yep.
Very nice talk. I really enjoyed. I have a question to bactobolin A. Actually, the last part. You were testing it on E. coli and Mycobacterium smegmatis. I saw it. The original structure is from Thermus thermophilus. Do you plan, or have you also tried something like a more serious bacteria? I mean, Staphylococcus aureus, where we indeed need new antibiotics, or do you have some opportunity to do so?
Right. So we haven't done it, but it is known in the literature. So bactobolin A is the most potent member of the family, and it was tested against various strains, including Staphylococcus aureus in some of the resistant mutants. So it is active. It is active against both gram-positive and gram-negative bacterium. So this is exciting. The challenge, but the real challenge I see is it's hitting the eukaryotic ribosome, because it binds within a relatively conserved site of the peptidyl transfer. And so we have to really walk around the molecule and see whether we find a lead and therefore an opportunity to get a selectivity. So this is really the fundamental issue I see right now. Okay. But it is active against some of these nasty strains.
Good. Okay, super. Thank you very much.
Thank you.
There's another question in from Lucas Meyer. Which project, from the perspective of the principal investigator, that's you, Jakob, was and perhaps is mentally the most exciting one?
Oh, Lukasz is putting me on spot. I don't know how to answer this properly. All of these, as you saw, had challenges, and it was fun to work on them. I really don't wanna rank them. Sometimes you have a wonderful idea that doesn't pay out at the end in the way you imagined, but I don't think so. I'm gonna tell to Lukasz that I really have enjoyed all three of them. All presented different challenges, and the important thing for me was that it was challenging, non-trivial, and so I'm happy with that.
It's a very political answer, Lucas.
I know.
Or Jakob, well done. And of course, the students involved in the three projects would like to hear that theirs were the most interesting, but you can't ever say that, so I understand. If I could just ask a very quick question, and you might want to find the slide to make it easier for people to understand what I'm asking. It was in the first Golan section where you were doing the metathesis, where at one stage with the Vega grubs, you went back to the starting material, they're just like there, but when you oxidized it, you then found it exclusively went to the tricyclic system you wanted. So, I mean, you kind of mentioned, oh, this must be due to some conformational change from the silyl-alenol ether versus the alpha-beta unsaturated system, and then it exclusively gives that. Have you modeled that reaction?
No, we didn't, but it is sufficient to take, at this point, really model in the hand, because if you're referring to this oxidized form, this would be a bread type olefin. So you would make a bicycle, if you imagine this bicycle, and it would have a double bond over here.
Yeah.
So that is, you would break your model set, try to make that. So we were pretty confident that this change will make the difference.
Okay, no, great. Good. So maybe with the interest of time, we'll stop there if that's all right. Jakob, well done again.
Absolutely, thank you.
Vladimir, over to you.
All right, thanks, Pat, and thanks, Jakub, for a great presentation. And next speaker is Veronika Nováková from Faculty of Science in Hradec Králové, which is also part of Charles University. So, Veronika, please, floor is yours. You are
okay. Can you hear me?
Yes.
And can you see my screen?
Yes.
In a proper way, yeah?
Perfect, yes.
Perfect.
Thank you. So thank you for a kind introduction. My name is Veronika Nováková. I'm from the faculty, from Charles University, Faculty of Medicine in Faculty of Pharmacy in Hradec Králové. And I would like to share with you the research done in my team in the field of ASA analogs of phthalocyanins. Phthalocyanins have been accidentally discovered in the beginnings of 20th century, but the structure was formed later. And they are closely related to porphyrins, which are present in biological materials such as heme or chlorophyll. But phthalocyanins themselves are completely organic synthetic dyes, and thanks to their quite simple scale of synthesis and very intense color, they are widely used as dyes in many different areas. Our ASA phthalocyanine group is interested in ASA analogs of phthalocyanins in which benzenes are replaced with pyrazines. And in my talk, I would like to give you a brief overview about their synthesis, about their photophysical properties and their applications for which we are developing our derivatives. So let's start with the synthesis. These very nice macrocycles arise from quite simple molecules. So the first task is to prepare the key intermediate, which is dichlorosubstituted pyrazine dicarbonitrile. This key intermediate can be prepared by condensation of diaminomalonitrile with oxalyl chloride, and subsequent chlorination give us this key intermediate. And then the following procedures depends on the type of peripheral substituent.
Yeah, I'm afraid, Veronica, do you hear us? We lost you.
Yep, now she just paused there.
Yeah.
The same for me.
Case when peripheral groups are connected via CC bond, we are typically using cross-coupling reaction, such as Sonogashira or Suzuki cross-coupling reaction. And some of the examples of the reactions are shown here. Finally, the precursors with peripheral groups connected through CC bond can be prepared also by condensation of vicinal diketones with diaminomalonitrile. This is a unique procedure for these pyrazine analogs. It's not possible to use them for synthesis of phthalocyanins precursors. Vicinal diketones are prepared, for example, by acetylene condensation. We are using either catalysis by potassium cyanate, or we are using thiazolium salts as catalyst, which is then followed by oxidation by copper-1 generated in situ, and finally condensation with diaminomalonitrile give us the required precursor. Alternatively, it's possible to use, we are using Grignard reagents, again with oxalyl chloride give us diketone, and again condensation with diaminomalonitrile may give us such precursors. Once we have a suitable precursor in our hands, it's used for cycle tetramerization to target macrocycle. And it can be done by two different methods, either by Lindstedt method or by template method. The first one uses alkoxide as the initiator of the reaction. It attacks nitriles group and activates the pyrazine for the attachment of another molecule. Then with the third one, fourth one, and finally it is close to macrocycle. The template method, in the template method, we are using some metal which serves as a template, and it enables getting 4 pyrazine units close to each other, and as a consequence they form macrocycle because it's thermodynamically more stable. In most of our applications, we need to prepare low symmetrical phthalocyanins, which is typically done by statistical condensation starting from 2 different precursors, A and B, and again either by Lindstedt method or by template method. We get a statistical mixture of 6 congeners which have units A and B combined in a random way. And from this mixture, we separate the desired congener. It's sometimes quite challenging because metal macrocycles typically tail on silica. But according to our experience, if the central metal is removed from the center by some acids, we get a metal-free derivatives. Their separation is usually simpler, let's say. So we isolate the desired congener from the mixture. And in next step, we may coordinate central metal back to the center. In one of our projects, we succeeded even in isolation of positional isomers, which is quite rare. Only about 3 papers mention successful isolation of these positional isomers. They arise if you are cyclotetramerizing unsymmetrical pyrazine, unsymmetrical precursor. And the funny thing is that we were not planning this project at all. We only borrow one of our compounds to my colleague for some testing reaction with tetrazoles. And he observed some unexpected elimination of nitrogen. So we studied this more in detail. We enlarged the series and used the precursors for the macrocyclic synthesis. And it resulted in a very nice ChemCom paper. The title of my talk was shedding some light on azaphthalocyananes, and why? Because if you shine a light on phthalocyanins and their analogs, they are able to efficiently absorb the energy of light and get to excited state. And from this excited state, they can relax back to the ground state by several mechanisms. And from phthalocyanins and analogs, there is a fluorescence and the generation of singlet oxygen being the 2 most often relaxation pathway. And probability by which relaxation pathway the molecule relax is based on some structural factors which we studied in detail. So, for example, there's a rule of central metal. It's based on a so-called heavy atom rule, and this rule said that introduction of heavy atom to the molecule emphasize the intersystem crossing. Once the molecule gets to the triplet state, it can transfer the energy to other molecules being in triplet state, such as molecular oxygen, which leads to production of singlet oxygen. There's also the effect of macrocyclic core. We have described many analogs of phthalocyanins and the effect of these different macrocycles on photophysical properties. And finally, probably the most important is the effect of peripheral groups, because if you introduce a donor of electrons to the periphery, there is a very efficient electron transfer to the macrocyclic core. And in such a case, the molecule relaxes through so-called intramolecular charge transfer. And the other pathway does not have a chance to occur. So such molecules are not fluorescent, do not produce usually singlet oxygen. So based on our experience, we are now quite familiar with these macrocycles, and we may design our derivatives for specific applications. And now please let me briefly explain the 3 applications for which we are developing our compounds. So let's start with photodynamic therapy. It is a modern or alternative method of cancer treatment. It's based on 3 components, molecular oxygen, light, and a drug called photosensitizer. And all these components are harmless. Only their combination leads to photodynamic effect, which is based on a production of singlet oxygen, which then leads to cancer cells death. Great advantage of phthalocyanin is the fact that these compounds are able to absorb red light, which penetrates deeper to the tissues. So the activation of photosensitizer in human body is efficient. In the field of photodynamic therapy, our group, the most of the effort in our group was done with phthalocyanins themselves, but due to structural similarity, we believe that pyrazine analogs should have some impact as well. Unfortunately, in all of our direct comparisons with phthalocyanins, the pyrazine analogs failed, even if we get quite nice EC50 values. The phthalocyanins were even better, and it was true both for compounds having rigid peripheral groups and or flexible ones. By the way, the rigidity of peripheral substitutions was found to be the key structural factor which produce highly efficient photosensitizers. We have also investigated why anionic compounds typically suffer from worse PDT activity than the cationic ones. And we found out that it's a combination of several factors. For example, aggregation at lysosomes, because there is a low pH, and at this low pH, the carboxylic groups are protonated, and as a consequence, these compounds, there are no repulsion forces and compounds aggregate, which is undesirable property. These anionic compounds have stronger binding to serum proteins and have different localization, relocalization after irradiation. So all these factors contributes to the fact that anionic compounds are always worse in PDT than the cationic ones. So pyrazine analogs are not optimal for photodynamic therapy, but when we studied pyridine ASA analogs of phthalocyanins with the rigid peripheral groups, we found out we developed a compound with extremely nice EC50 value of 4 nanomolar, which is one of the best photosensitizer ever. And this is especially interesting from the point of view that commercial available photosensitizers used in practice, studied under same experimental condition, had worse activity than our compound. But let's get back to pyrazine analogs. So they are probably not optimal for photodynamic therapy. So let's have a look on their impact in a fluorescence sensing application. This application is based on the fact that macrocycle serves as a signaling moiety, and on the periphery there is a donor for electron transfer, which is a part of recognition moiety. And in this free form, the electron transfer quenches efficiently the excited states, so the molecule is completely non-fluorescent. And upon meeting a sensitive analyte, which binds to the recognition moiety, the quenching process is blocked, electron transfer cannot occur, and it leads to restoration of fluorescence, so the compound is, the sensor is switched on. And blocking of this quenching process can be done either by protonation of donor, which give us pH-sensitive compounds, or by coordination of a cation to the azacron moiety, which give us sensors for metal cations. Typically, once we prepare a series of sensors, we do some titration experiments with various analytes, and there are no changes in absorption spectrum, but we, in the sensitive cases, we observe a steep increase of fluorescence. We determine quantum units of fluorescence, calculate the association constant, and discuss usually the selectivity and sensitivity of the sensor. On this slide, I give you a few examples of our pH sensitive derivatives. The first group is based on protonation of using amine as a donor for electron transfer, and it's protonation which leads to switching on. In this case, we proved, for example, that it's necessary to have a linker between donor for electrons and the macrocyclic core. We also studied the possibility for tuning the pKa of amine. And we described also the effect of the distance of donor from the acceptor of electrons. So we can easily play with many factors to get the optimal sensor. Another possibility is to use phenol-phenolate pair for switching between on and off states. Because phenolate is extremely strong donor for electrons, which keeps the molecule in off state. And by choosing proper substituent X and Y, we can tune the pKa of this moiety. We are able to prepare compounds sensing from strongly basic up to acidic pH range. The disadvantage of this compound is that they are lipophilic, they are not soluble in water. So in all these cases, we are using liposomes or microemulsions as delivery systems. pH sensitive compound can be used also in sensing of CO2. In this case, we took one of our pH sensitive compound. It was immobilized on polyurethane matrix along with a strong base. And this base keeps the sensor in off state. And once the CO2 comes to the sensor, it goes to protective gas permeable protective silicon membrane to the sensing layer, and it causes acidification of protonation of phenolic group, which results in steep increase of fluorescence, and fluorescent sensor is switched on. In this project, it was done in cooperation with Sergei Borisov from Austria. We developed a device based on fiber optic system, which was compatible with a commercial fluorimeter. And this sensing device showed very stable, reproducible response to CO2. So we were extremely lucky about this application of our compounds. Regarding the sensing of metal cations, with this topic we started almost 10 years ago. In the first project, we described some interesting supramolecular arrangement. We studied stoichiometric binding, and we proved that the compounds work in water as well after in embedding to silicon nanoparticles. And in the next years, we focused on improvement of their selectivity and sensitivity. Firstly, we looked on the impact of size of the exochrome, and later we described that rigidity of recognition moiety can give us compounds which are really extremely selective. Unfortunately, in the case of tweezer arrangement, there we observed that the quantum yields of fluorescence in the on state are quite low. And it was because we have two donors for electrons. So upon activation, we still do not have the maximum possible value of the quantum yield of fluorescence. So in our last project on this topic, we design and prepare derivatives with only just one donor for electron transfer. And the strength of binding was achieved by some Lariat ethers or some additional crowns. And all these derivatives show extremely nice selectivity and sensitivity toward desired analytes. And interestingly, in this project, we also observed that there is a big role of counter anion used in the titration experiments. And we suggested that it nicely correlates with the Hofmeister series, so that the effect somehow correlates with the caltropic ability of these counter anions, but still there was extremely high selectivity toward thiocyanate salt. So we investigated this in more detail, and we found out that our compounds behave as ditopic sensors, combining size fit recognition of cation with the coordination of thiocyanates to the central zinc atom. And since the sensitivity was extremely high, we introduced these compounds to biologic experiments. And in these pilot experiments, we proved that fluorescence increases even in cells upon addition of thiocyanates, which was not observed for the negative control experiments. Further, we have developed a simple method for quantification of thiocyanates in biological samples, for example, in saliva, which is quite important for smokers who have increased levels of thiocyanates in saliva, and we got biologically relevant values which confirm that the method works well. So these promising results force us to continue in this topic further. So we plan to develop compounds which will be suitable for bioimaging. The last application for which we are developing our compounds is quenching of fluorescence in DNA probes. This topic would deserve own lecture, so I will only briefly summarize it on this slide. In this case, we are using compounds with more than 4 donors of electrons, because such compounds are completely quenched, and they are able to quench fluorescence of other compounds, either by resonance energy transfer or by static quenching. The great advantage of our derivative is that they have panchromatic absorption spectrum. They cover, they can quench the fluorescence of all commercial available fluorophore, which is highly advantageous. We have developed methods of binding our compounds to oligonucleotide probes, either as monolabeled probes. We have methods for preparing double-labeled probes, having quench on one side and fluorophore on the other side, and for triple-labeled probes. And we proved that these probes are usable in PCR assays. To conclude my talk, I hope that I convinced you that ASA analogs of phthalocyanins enable wider synthetic possibilities for precursors than parent phthalocyanins precursor. Unfortunately, they are not optimal for photodynamic therapy, but they have great impact for applications involving electron transfers, such as sensing fluorescence sensors and quenchers of fluorescence. Finally, please let me thank my coworkers, especially PhD students and master students who did most of the experimental work. And I would like to thank also my coworkers from Czech Republic and abroad. Thank you for your kind attention.
Monica, super. Many thanks for that. And again, if the session is open for questions and answers. Questions and then answers. Here we go. And Basha, again, in photodynamic therapy, have you quantified the reactive oxygen species?
Yes, sure. We usually we do this. We have the numbers in the paper, but sorry, I don't know the values at this moment. For all our derivatives, we determine the quantum yields of singlet oxygen in solution, in organic solvents or in water, and then we subject the compounds to in vitro experiments or in vivo, and the biologists do all advanced experiments for us.
Great, thank you. Philip, you're next. You have your hand up, please.
Yeah. So Veronica, thanks for your nice contribution. I would have a question concerning your really initial slides you shown us, and which is also related to my work I will present later on. And it concerned the condensation of 1,2 diketones with diaminomallonitrile. Did you also try to perform this reaction with some really electron-rich aldehydes or sorry, ketones? Because so far I have seen you have just some alkyl substituted and some benzyls, in fact, substituted benzyls. Yeah, this is the case, exactly.
Yeah. Do you mean some?
I don't know, thiophene based and or maybe even stronger electron donating moieties instead of phenyl rings?
No, actually we did not need that.
Okay, because this is quite problematic even for us. Yeah, I just wanted to know whether you have some experience. Okay,
I cannot help you.
Okay, Vladimir.
Yeah, I wonder about the synthesis of microcyclization about these cyanines with different units. So you said that you are using the statistical approach. So can you kind of preferentially, prepare, for example, like with 1 unit or 2 units? Are there some tricks about that?
Actually, you can play with the ratio precursors. So if you use 1 in excess, then you may support the formation of some desirable one. And to be honest, nowadays we intensively work on some controlled way of preparation of low symmetrical phthalocyanins. So nowadays we have 2 different projects with 2 different PhD students who focused on selective synthesis of low symmetrical compounds, because this is extremely important and interesting for us. So let's hope that in future I will tell you more about that.
All right, thanks.
Great, thanks, Vladimir. Michael, over to you.
Thank you. Veronica, thank you for a very nice talk. I just have a question regarding the conducting properties of your materials, because phthalocyanins are known to be highly conductive. And what about the pyrrosines? Have you considered to use them as conductive layers?
Actually, we have so many things to do that we did not focus on conductivity of these materials. But yeah, yes, I know when we have a conference on phthalocyanins, which is quite full of experts on the phthalocyanins, there are many very nice talks about conductivity of these materials, but we are not focused on this.
Okay, sure. Thank you.
Great. So Veronica, we might leave it at that just in terms of time. Thank you again for an excellent presentation. Vladimir,
thank you for inviting me.
You're welcome.
All right. So our next speaker is Michal Kohout from University of Chemistry and Technology in Prague, and he will tell us something about chiral hybrid organic and inorganic materials. So please, Michal.
Okay. Thank you very much, Vladimir. Do you see my screen, please?
Yes.
Yep.
Perfect. Okay. Many thanks for the kind introduction, and I'm very honored to have this opportunity to talk today at the highlights of organic chemistry in Czech Republic. Though I'm not going to talk very much about organic chemistry, I'm going to talk mainly about materials and hybrid materials, organic and inorganic systems. Hopefully it's organic enough. Okay, just to give you a brief overview of what we are doing in my work group, we deal with liquid crystals, and typically we actually work with some photosensitive compounds. Also, we work on light tunable materials, and then we are focusing on some hybrid materials for liquid chromatography, for chiral separation, and also for organocatalysis. So today I'm going to talk about two different things. The first is the chiral synthetic and semi-synthetic selectors for liquid chromatography. Actually, just to note about chirality, we have seen already several talks about chiral systems and chiral molecules. I just would like to mention that we are also chiral, all of us. And since the microscopically chiral objects can talk to each other, for example, as hands. You can shake the hands. You have different possibilities to grasp your hand and to hand your hand to somebody else. The same do the molecules. The molecules talk to each other. The molecules can grasp each other in different ways. And we can actually distinguish chirality on the object level. So we have chiral objects which are easily to be distinguished. For example, these shells of a sea snail. But when we go to the nanoscopic level, to the level of molecules, then it is very difficult to distinguish between the enantiomers, although they differ very much in their properties. So we know that the properties of the S enantiomer and R enantiomer of, for example, a very easy compound like cathinol have different properties. So generally, we are facing the issue that we need to distinguish the enantiomers to have the properties we would like to deliver. And the thing which enables us to do that is enantioselective separation. For that, we have several methods. We can use so-called indirect approach, where we take our racemic mixture and we use chiral derivatizing agent, and we create a pair of diastereoisomers, which then can be easily separated using crystallization or achiral chromatography and some other methods. We can also use so-called direct approach, where the same pair of enantiomers is interacting with a chiral selector, which is either in the mobile phase or it is immobilized on some stationary phase. And then we can form just diastereomic associates. So there is no covalent bond, but this is enough. The diastereoisomeric associates differ in their physicochemical properties, and we can separate these entities. The ideal approach, from my point of view, is to use chromatography. And the thing is that we need chiral selector, which is immobilized on some solid support, and then one of the enantiomers fits perfectly into the binding pocket of the selector. So it is this lock and key principle, which we know from the biochemistry and biology 1 from the lectures. And then we create a more stable diastereomeric complex. And for the other enantiomer, we have the same selector, and the enantiomer does not really fit into the binding cavity of the selector. So there we create less stable diastereomeric complex, and we can separate these entities, of course. So when we have, let's say, higher binding constant and smaller binding constant, then we can reach to the separation factor alpha, which gives us the difference in energy of binding. And this is principally the thing which we need for separating our enantiomers. One of the organic selectors which are used for chiral separation are selectors based on quinine and quinidine on cinchona alkaloids. There is easy modification of this scaffold, and this creates an ion exchange selector, which is actually very efficient in enantiorecognition of organic acids. Here we have an example of N-protected amino acid. And you can see that here we have ionic interaction between the charged kinucleidine ring and the protonated carboxylic acid of the amino acid. This is due to the environment. This is due to the mobile phase, which is weakly acidic. And under the weakly acidic conditions, we have these two charges. And this is the primary interaction we see between the charged selector and the charged analyte. Subsequently, there are additional interactions, the hydrogen bonding, for example, pi-pi interactions, steric interactions, which are responsible for the actual enantiorecognition, which are responsible for more tight binding of one of the enantiomers and less tight binding, less stable diastereomeric complex of the second enantiom. Since we are talking about charged stationary phase and charged analyte, we need some reasonable buffer which we use to liberate the analyte from the charged stationary phase. Otherwise, we won't be able to do that. We are playing a little bit with this structure, with the structure of the selector, and we actually studied two possibilities of immobilization of such selectors on stationary phase. We used the quinine double bond, and we also introduced a triple bond on the carbomoyl moiety to be able to play with the site of immobilization using either mercaptopropyl modified silica or azidopropyl modified silica. I will just give you one example of a selector, which is here, and you can see that from switching from quinidine to quinine, we can also switch the elution order of the enantiomers of our analyte. This is quite important because if we would like to go for controlling, for example, enantiopurity of some API, we would like to have our impurity or the second enantiomer, let's say, enantiomeric impurity to be the first peak in our chromatogram to be able to detect it in very tiny amounts. So this is the possibility how we can do that. We can use so-called pseudo-enantiomeric stationary phase to change the elution order of the analyte. Currently, we would like to explain what is going on during the chiral recognition process. What is actually the force which is responsible for elution order of our analytes, the sequence in which the enantiomers elute from the chromatographic column. For this experiment, or for this study, we selected dinitrobenzoyl beta-phenylalanine and our chiral pack QNAX and chiral pack QDAX, the chiral columns based on quinine and quinidine. And what we do see here is that we actually experience different types of interactions for different enantiomers. It's clear, but here, using this in silico approach, in silico dynamical approach, we can actually quantify the enantiomeric interactions which are responsible for the enantio recognition process. So this is something which gives us the opportunity to say we experience that many pi-pi interactions, that many cation-pi interactions, that many hydrogen bonds, and therefore we observe this elution order chromatographically. So this is one of the points where we are actually taking into account also in silico modeling and NMR and all these other things. So this is what leads us to explaining the chromatographic processes. I will just show you some additional selectors. These are cation exchangers, which are very nice systems for enantio separation of chiral bases. You have, for example, mefloquine as one of the antimalarials which are being used, and we can separate mefloquine very effectively with these cation exchangers. What we did so far, we played with the lateral substitution. We exchanged original utilized chlorines for methoxy groups, and you see that if we change chlorine for methoxy, we have a little bit higher enantio selectivity and a little bit higher resolution of our target analyte, which was mefloquine in this case. And this is because we are changing the nature of the central aromatic core of our selector. So we are forcing it to be pi base, which is then better in pi-pi interactions with the pi acidic core of mefloquine. So this is the principle how to enhance the interactions between the two entities. We also played a little bit with the linker unit. We used either sulfur or triazole, as you can see here, which we studied in this last publication, and we have seen that there is some effect of the size of the linker. So we can actually enhance the enantio recognition properties of the selector using the bulkier linker unit. Joining together the quinine moiety or the cinchona moiety with this cation exchange unit, you can eventually come to the silica ionic system, The Zwitterionic system has been patented already in 2008, and we have finished the technology transfer in 2012, so they are commercially available from 2012. These are systems from Lindner Group, and since we are using quinine and quinidine again, we have the possibility of reversal of evolution order of amino acids and peptides, particularly because these solvents were developed for enantiomer recognition and enantiomer separation of non-derivatized amino acids and peptides, which is quite difficult to actually achieve in a different way. But these systems are very efficient also in retaining other polar uncharged molecules. We have studied their properties also under supercritical fluid chromatography conditions, for example. And you can see here that we have used N-acetyltryptophan and methylquine as analytes. And when you use the classical chiroplac UNAX column with an ion exchanger, you can nicely separate this analyte in 50 minutes, which is quite long. The same for the cation exchanger, you can separate methylquine in 30 minutes, which is also quite long. But if you use the Zwitterionic ion exchanger, you see that the resolution is better and the retention time is much shorter. So this is actually the winner. We then utilize that also for enantiomer separation of drugs, and we were able to achieve enantiomer separation of parent drug and its metabolite in one run very nicely. You have separation of both, and the metabolite is separated a little bit better because it's more polar. Recently, we are focused on chiral resolution of dipeptides under buffer-free conditions and in reversed phase. So that means that we are using just methanolic mobile phases, so methanol water mixtures without any buffer, and we can nicely separate dipeptides using slightly modified structure of our original selector. And this leads to harvesting pure dipeptides without any necessity or without any subsequent purification, which is typically necessary when you use buffered systems. Okay, and now I'm going to focus on chiral photosensitive liquid crystals in magnetic nanocomposites, so a little bit different branch of hybrid materials. First, I would like to mention that the reason why all of us are here are liquid crystals, because as you can see in these pictures, small fragments of DNA or short fragments of DNA form nice liquid crystalline phase, and also phospholipid bilayers are liquid crystalline. So these are liquid crystals. So quite a lot of biological systems are actually liquid crystalline. But today I'm not going to talk about them. I'm going to talk about thermotropic liquid crystals. And the reason is that they were discovered in Prague. Actually, in 1898 at the Charles University, the first node of liquid crystals comes from Prague. Then there were other groups working in liquid crystals of various shapes. Here we have the bent cores and also the disc-like liquid crystals. Just a little bit about the self-assembly of these molecules, because that's the key property which enables practical applications of liquid crystals, that the molecules can self-assemble into specific structures which are sensitive to external stimuli, such as electric field, light, or magnetic field, or pressure. So actually, when we start from solid phase and we heat up the solid phase, we can eventually arrive to smectic C phase, where the molecules are tilted and they are oriented in layers. The same applies for smectic A phase, where we still have the layered ordering of the core system and the molecules are just orthogonally oriented. And nematic phase, where the molecules are oriented just along their long axis, short axis for this cortex, without any other arrangement. And finally, when we increase the temperature even more, we reach isotropic liquid. So basically, one of the interesting features of liquid crystals is that if we introduce a photosensitive unit, we can also work with light as a stimulus and organize or change the organization of the mesophase by light, which is quite interesting because we can then control the organization remotely. It means that we can switch the nematic phase by application UV light into isotropic liquid, and then the isotropic liquid relaxes back to the organized state, either by thermal relaxation or by applying visible light. So this process is fully repeatable, and we can play with the system and organize and disorganize it. So that means that we can actually be in state 1 and state 2. So this is the basic for logic, of course. Okay, what about the design of our molecules? The design started from these compounds, which we published in 2016. They had relatively nice properties, including some properties which would be theoretically applicable. And we thought, okay, what is going to happen if we introduce a photosensitive unit in here? Actually, we still had very nice mesomorphic properties, and also we were able to organize the molecules within the mesophase by UV light, which is shown here, because if we apply the UV light, we switched the mesophase off and we obtained just isotropic liquid. This is a masked area where you can see that the mesophase was preserved, and this is the borderline area where it is a little bit unstable, of course. When we switched off the UV light, the mesophase grew back, and you see that this was the unmasked area and this was the masked area. And when you repeat the experiment, so this is the repetition, you see that again, the mesophase was switched off. We are in isotropic state. Here is the original mesophase. And when you switch off the UV light, the system regrows back, and now it is completely healed. So the process works perfectly fine, and we are able to modulate the organization of the liquid crystalline compounds within the mesophase. Just moving a little bit onwards, then we thought, what is going to happen if we immobilize our materials on nanoparticles? Because we knew that there were systems where the photosensitive compounds were bonded to nanoparticles. There were systems where chiral materials were bonded to nanoparticles, creating chiral mesophases. And there are examples of chiral photosensitive dopants into liquid crystalline hosts. But we decided to go a little bit further, and we thought, okay, if we are able to modify magnetic nanoparticles, then we can create systems which will be magnetic. They will be photosensitive due to the presence of a photosensitive unit, and they will be chiral due to the presence of the center of chirality. So our goal was to create this 3-in-1 dopant into liquid crystalline matrix. To do so, we synthesized the linker towards the nanoparticle, which was based on phosphonic acid, the photosensitive core. So this was the first precursor. And the second precursor, for this purpose, we used lactic acid, which is one of the cheapest sources of chirality ever. And we created such chiral unit, which was then connected to our photosensitive central core with the linker. And eventually we arrived to this series of materials, to this series of ligands, which we used for modification of magnetic nanoparticles. We modified the cobalt ferric nanoparticles using, at the end of the day, hexylphosphonic acid as a co-surfactant, because if we are not using the co-surfactant, then the aromatic units tend to lie down on the surface of nanoparticle, and we see actually almost no effect whatsoever. We actually see, or moreover, we actually see the aggregation of nanoparticles, quite fast aggregation. Even though we used hexylphosphonic acid in this case, the colloid formed with liquid crystal host and our dopant was not very stable. So we thought, what can we do to stabilize it? And actually, my student back then, Anna, came with an idea that we can create a trimeric system. So then we discussed what is necessary to create a trimeric system. And after discussing that, Anna went back to the laboratory and started the synthesis, which looks like this. First, we needed to create the photosensitive core. And sometimes we prefer the shortest ways, you know, the shortest way used to be the better, or typically it is the best option. But in this case, it was not true, because the 3-step synthesis provided the product just in 8% yield, while the 5-step synthesis was a little bit better. And we obtained the product in, or this intermediate, in 31% yield overall, which is very good. But the last strategy with 6 steps afforded the intermediate in 44% yield. So actually the longest synthetic strategy was the best in this particular case. Then we actually synthesized also this linker towards the nanoparticle. Putting these fragments together, we achieved to have this nice dendritic system, which at the end of the day was actually functionalized with the chiral unit. And after the protection, we had this very nice ligand. Having the ligand in hand, we tried to modify the nanoparticles using tolyylphosphonic acid, this time as co-surfactant. And as you can see, in this case, we were successful, and we had very nice homogeneous arrangement of the modified nanoparticles. So there was almost no aggregation. And also when we mixed it together, when we mixed the dopant together with our homemade liquid crystal, you see that here is the polarizing optical microscope texture, and you see that the texture is perfectly smooth. There was actually no aggregation in there, so the system was perfectly stable. Subsequently, we asked Torsten Hegman from Kent to measure either induced sarcoidal dichroism to see whether the system is chiral or not. And this is the liquid crystal used, which is a chiral. But here we can see the induced sarcoidal dichroism measurement from the nematic phase and from the smectic phase. And we actually see nice effect, and we know that the system is chiral. So this proves that the chiral induction is there. Subsequently, we tested the photosensitivity of the material, and you see that again, this is the picture before the UV light application. Here we see disappearing nematic phase under the UV light. And when we switched off the UV light, the mesophase grew back instantly. So basically, this is proof that the system is also photosensitive. I'm not showing the magnetic data. The reason is that there is quite a lot of magnetic measurements along this line, because surprisingly, the magnetic response of the material was higher than we expected. Because normally you just dilute the magnetic nanoparticles with the organic material. But in this particular case, we observed some effect of coating, which increased the magnetic response of our nanomaterial. So the mixture with liquid crystal was also magnetic. So finally, we had our dreamed 3-in-1 dopant. We had a system which is magnetic, photosensitive, and chiral at once, after doping an achiral liquid crystal with our nanomaterial. So this was the goal of this study, and we were successfully reaching it. And I guess that this is one of the possibilities how to create new type of dopants with really interesting functionalities. At the very end, I would like to thank all my colleagues from the University of Vienna, all former and current students who do actually all the job. I would like to thank Torsten from Kent State University and the guys from Institute of Physics and all the other people from different universities whom we cooperate and work on different aspects of chirality and hybrid materials. I would like to acknowledge also Czech Science Foundation for financial support, and thank you very much for your kind attention.
Great, thanks, Michal. And just waiting to see if there's any questions coming up in either the chat or Q&A or attendees. I don't see any so far. Oh, Vladimir.
Thank you, Michal, for nice lecture. I have a question about concerning the first topic, the chromatography. So can you actually predict how your exchanger will behave, for example, based on just studying basic pairs of supramolecular properties between this molecule, which is part of the exchanger, and the chiral molecule which you want to separate?
Basically, I think that we will be able to do it using this type of in silico chromatography. That is one of the options which we now trying with the colleagues from Perugia, and they are very good in that. And I believe that once you have the selectors fitted into the sandbox, fitted on the silica layer, you can pass any analyte of your choice, and you can actually I would say even quantify the interactions between the selector and the analyte, and then you can say, okay, here I have enough interactions for efficient separation, so this will work, and this selector is the best for that.
Okay.
Maybe a related question, Michael. Are there any classes of compounds that are obviously enantiomers that cannot be separated by chiral chromatography? Or what classes of compounds are difficult?
Difficult are heterocyclic systems which are small. So tetrazines, chiral tetrazines are quite difficult typically, and also the polar substances. Therefore, we work with ion exchangers, because when you take, for example, basic pharmaceuticals such as propranolol and some other beta blockers, you cannot actually separate them very efficiently with chiral stationary phases based on polysaccharides. And polysaccharides are the best in the market these days.
Yeah.
But they fail with these slightly polar systems. So sometimes there are obstacles also for the polar compounds and charged compounds in particular.
Very good. Thank you. Now, I don't see any other questions coming in. So maybe in the interest of time, we might just keep moving on ahead with the program, if that's okay. Michal, thanks very much.
Yeah, thank you.
Over to you again, Vladimir.
Yep. Thanks, Pat, and thanks, Michal, once again for a lecture. So next talk will be delivered by Lucie Brulikova from Palacký University, Olomouc, and she will tell us something about small molecules which act against drug-resistant pathogens. So please, Lucie, for yours.
Thank you very much. Can you see the sharing on my screen?
Yeah.
Presentation. Okay. First of all, I would like to thank the organizing committee for inviting me to share with you results of my research team that is mainly focused on the developing of novel antimicrobacterial agents. In the beginning, I would like to briefly explain why we decided, or I decided for this research. So I will briefly summarize some important facts about tuberculosis. Tuberculosis is a communicable disease that is caused by bacteria, Mycobacterium tuberculosis. Tuberculosis is one of the leading causes of death worldwide, and until the coronavirus pandemic, TB was the leading cause of death from a single infection agent, ranking above HIV. And moreover, TB is also the leading cause of deaths related to antimicrobial resistance. The alarming fact nowadays is the fact that MTB latently infects about 1/4 of the world's population, what is about 2 billion people. And these people are not ill with the disease and cannot transmit it, but this number is really alarming. Other tuberculosis, curable and preventable, the alarming increase in drug-resistant cases dramatically complicates the treatment, and reasons for re-emergence and spreading of tuberculosis are mostly emergence of resistant strains like multi-drug resistance and extensively drug-resistant strain. Then appearance and spreading of hypersusceptible populations like people with HIV or homeless people. And finally, in developing countries, the treatment is insufficient or completely lacking. For this reason, many researchers across the world are dealing with development of novel, better anti-tuberculosis agents. And we can target special enzymes or molecular target at 2 levels. I would like to show you some briefly again what is maybe the most important in the developing of novel anti-tuberculosis agents. Here you can see the Mycobacterium tuberculosis and some specific molecular targets at the level of this bacteria. And you can see that we can target cell wall, we can target redox homeostasis and oxidative phosphorylation, for example. All these targets are vital for bacteria. So when we inhibit these targets, we can kill bacteria, and then we can target some special processes at the level of the host. So we can somehow help your immune system to fight against bacteria itself. Today, I will talk about 2 targets. I will talk about ATP synthase, what is the most important target for me, and then I would like to also talk about metalloprotease ZMP1. I will explain it later. What does it mean exactly? So now let's take a look at the first target, mycobacterial ATP synthase. As you can see from this picture, this enzyme is really complex structure. This consists of several units and subunits, and I would like to show you also some movement. And please pay attention to this part. The rotating part is subunit C, and the blue one here is static subunit A. I will talk about this part of enzyme later in connection with our compounds. So this part is a rotating subunit, and this is static subunit. And this enzyme is very important in the development of drugs against drug-resistant mycobacterial strains, because this target is really known, and the first target discovered, the first drug discovered to target the ATP synthase was bedaquiline. This was compound approved by FDA in 2012, and this compound started a lot of attention of chemists or medicinal chemists because this compound was about after 40, maybe 40 years after the last compound efficient against tuberculosis. And despite efficiency of bedaquiline, bedaquiline has several drawbacks, such as the long half-life, high lipophilicity, low solubility. And for this reason, many researchers decided to find novel analogs that can target ATP synthase, mycobacterial ATP synthase. And what is the most important fact? That there are significant structural differences between mycobacterial ATP synthase and human ATP synthase. So inhibition of mycobacterial ATP synthase is considered as a relatively safe approach in treating drug-resistant TB cases. Now I will show you what we are dealing with in my research team. We decided to study more compounds and this molecular target. You can see the overview of our project. Some times ago, we decided to study scleramides. This group of analogs was discovered or published in 2017 by groups of authors in EuChemS, and this is really good group of compounds because scleramides were found to target subunit A, the static part. And then I would like to talk very briefly about pyrazole or pyrazinones because these compounds were discovered during virtual screening. They are described that they are targeting ATP synthase, but the exact target within ATP synthase is not clear to date. Then we are dealing with pyrimidine analogs. This compound was described in 2012 by a group of authors, and these analogs were described that they are target subunit gamma. This is part of the enzyme, and we have some doubt about the target within ATP synthase. So we decided to investigate this group of analogs. And then we are dealing with biphenyls and thiazolidinedione derivatives that were found to target subunit C. This is a very brief overview, and I would like to talk in more details about scleramides. Scleramides were discovered in 2017. They were proved to be very active against drug-resistant cases, but I think that the SARS study that was performed by Tantri and co-workers in 2017 was not efficient, and we decided to extend the SARS study and to better explain the possibilities of these structures. During last 3 years, maybe, we synthesized about 80 compounds with aromatic moiety, with amine moiety and amide moiety. And as you can see from this overview, only compounds with aromatic moiety, it means that there is aromatic part directly bind to squaric cycle through CC bond. Only this compound of this series of compounds are the most active analogs, reaching the MIC 2 micromolar. We tested these compounds against Mycobacterium tuberculosis. We detected no acute cytotoxicity. We also tested all compounds against panel of bacteria and other pathogens, parasites, and we discovered the minor activity of some compounds belonging to the first group of analogs, minor activity against Leishmania donovani. We experimentally prove the molecular target with this subunit A. I will talk about it in more detail later. We raise continuous mutants and experimentally prove this target. We also tested or proved the accessibility of these compounds to beta-acylin resistant mutants. So what is a very nice fact that these compounds could be used for treating of drug-resistant TB cases where beta-acylin cannot be used, for example. And now I would like to talk about the synthesis in more detail. This is highlighting organic chemistry, so I would like to highlight some synthesis. We started with the reaction what was already discovered or described by Tantri and co-workers in 2017, and this reaction or the crucial or key reaction of this synthetic procedure was based on the CC bond formation in this step. Very briefly, we started with commercial available squaric acid that was converted to dichloro-derivative. Then the Friedel-Crafts-Steiber reaction was performed, and finally the substitution reaction with 2-aminomethylpyridine gave us analogs 1. But this case step turned out to be insufficient, or it wasn't useful for various type of modifications. So we tried to find some better synthetic approach, and we tried a lot of, for example, coupling reactions, and we discovered this Schlegel coupling reaction that was very useful for us. And thanks to this reaction, we prepared about 30 new analogs. This reaction started with dichloro-derivative that was converted to intermediate 7. Then it was reacted with paramethoxyethylphenol to give this crucial intermediate for coupling reaction. Then the reaction with various boronic acid gave us methoxy derivative that were converted to final compounds with substitution reaction with 2-aminomethylpyridine. So this reaction is the best one to date for what we can use for synthesizing of these type of analogs. Then we also synthesize. We also tried to do a reaction with lithium salt, and then in this procedure we started again from squaric acid, commercially available compound that was converted to dimethoxy derivative, and this derivative was reacted with in situ prepared lithium salt to give us this ketal. And this intermediate was reacted to aminomethylpyridine again to give us intermediate 13, and this intermediate was then deprotected to give us final compounds 1. So we tried a lot of synthetic approaches, but I think that reaction based on Schlegel coupling reaction is efficient and used for, and can be used for variable substrates. Then I will show you again very simple synthetic methods for preparation of amines. The second group of analogs, we started with squaric acid, converted to dimethoxy derivative, performed substitution reaction, and the second substitution reaction with amines, we got desired analogs 2. And the third group of compounds, amide derivatives, synthesis started again with dimethoxy derivative that was reacted with gas ammonia, and then amine group was reacted with carboxylic acids to give intermediate 16, and again last substitution step gave us amide 3. As I already mentioned, only the first group of analogs that analogs were the best from the tested libraries. And when we experimentally proved the molecular target, we also wanted to explain the behavior of our compounds using molecular docking, and to better explain the behavior. We docked our compounds into the model Mycobacterium smegmatis because the crystal structure for ATP synthase for Mycobacterium tuberculosis was not known, and but Mycobacterium smegmatis has a very high level of similarity, so we can I believe that we can use it for explanation. We studied the active site where these amino acids were glutamic acid, lysine, and asparagine, and this is gene that encodes subunit A. Maybe this picture on the right-hand side is more visible. You can see our structure here. That is bind to subunit A, this part, and here you can see rotating subunit C, And here is the bedaquiline. Bedaquiline is that standard approved drug against tuberculosis. This picture also can explain why our compounds are sensitive to bedaquiline-resistant strains, because very obviously our compounds target a different active site or different subunit of ATP synthase. And what is maybe interesting that employing the molecular docking, we found out that these compounds, or our compounds, target this subunit only in stage 2. So it means only in state where ATP is formed. Okay. Now I would like to move to the second group of analogs, pyrimidine analogs. I mentioned in the beginning that we started to investigate these compounds because I have some doubt about the molecular target described in the published paper. So I wanted to prepare some another analogs and study in more detail. In this project, we are in the beginning, we just synthesized some compounds, and now we are waiting for the biological activity results. But I can show you at least the synthesis of these compounds. Was started from commercially available amine and intermediate 19. We got nitro derivative 20 that were reduced to give intermediate 21 amine, and these amines were reacted with papered chloropyrimidine to give us the final compounds. And this chloropyrimidine was prepared from dichloropyrimidine, a commercially available compound. And as you can see from this scheme, we observed the formation of two regioisomers, but this desired isomer was in excess. This was the major one. We are, as I mentioned, we are waiting for biological activity testing results. Then we can move this project by substitution other positions and not maybe try to change the central part of molecule. So, and from this point of view, the last scheme, I also talk about pyrazolopyrazinones. Again, I will show only the synthesis, and we are waiting at this moment for biological activity results. Synthesis started with this ester that was reacted with dibromo derivative, converted to azide, cyclized to pyrazolopyrazinones that was reacted with amine or different type of amines in the presence of 3-methyl aluminium, then substituted or alkylated with benzyl bromine to give us compounds 25. Okay, now I would like to move to second target, ZMP1. And firstly, I would like to explain what should happen with bacteria when enter the lungs and are collected in the alveolar sacs. Bacteria are phagocytosed by alveolar macrophages to create phagosome that should fuse lysosome to create phagolysosome. And this complex is supposed to remove invalid pathogen that should lead to valence attenuation. However, Mycobacterium tuberculosis has an ability to block some of these processes mostly by the specific cell wall construction, and moreover due to so-called valence factors, where the special molecules that are formed by bacteria, and these valence factors are able to block some of the mentioned processes, and so block immune system response. And one of these bronze factors is ZMP1, what is zinc metalloprotease, and we are dealing with developing agent able to inhibit this enzyme, because we believe that the inhibition of this enzyme could lead to the discovery of some compounds that could be useful as adjunctive treatment. Some additive drug to drug target, especially the bacteria. This enzyme is essential, or was found to be essential for intracellular survival and pathogenesis of Mycobacterium tuberculosis. And it was found that this enzyme affects the phagosome maturation by a suppression of inflammasome activation. So we started to synthesizing some of these compounds, and I will show you 2 generations. Firstly, we were inspired by literature and prepare some compounds. And here you can see the most active analog from the first generation. And then we suggested another analogs, and these two compounds, 34A and 35A, were the most active one from those libraries. We prepared about 50 compounds, but you can see almost all of them, or these most active analogs, had similar results. They exhibited IC50 against recombinant ZMP1 about 20 micromoles, but it's not really the bad, but not best. So we would like to get the more active analogs. Synthesis was very simple. I will very briefly comment it. We started with thiazolidinedione. Then it's alkylated, hydrolyzed, and reacted with allyl hydroxylamine to get hydroxymates. And the second library was started with thiazolidinedione again. That was subjected to Knoevenagel reaction, then alkylated, hydrolyzed, and reacted with hydroxylamine. That means to get final compounds 32. And the second library was designed based on the results of the first library. You can see this picture where this is active site of ZMP1. You can see zinc ion, that is inactive site. And in front, you can see the standard inhibitor of a lot of metalloproteases, phosphoramidone. This is the gray structure, and you can see that nicely interact with zinc ion in the active site. And in back, you can see the black structure, what is our compound, and you can see that it doesn't interact with zinc ion, and this compound was found to rather occupy the entrance into the central cavity. For this reason, we designed second generation, where we suggested some analogs with longer peptide chain that could interact with the zinc ion in the central cavity. And very briefly, synthesis of some analogs, indole and pyrrole and indole moieties. It started from commercially available compounds, and this is again very, very simple synthetic procedure: alkylation, hydrolyzation, then reaction with hydroxylamines, and hydrolyzation of methyl ester, reaction with dipeptides, and hydrolyzation. Got these final compounds. As I mentioned, we want to reach some better results. For this reason, we started collaboration with Pavel Poliszczuk from IMTM, and he performed de novo design and suggested some molecules what we are synthesizing right now. And I'm really I'm interested why or how these compounds will behave, and how the theoretical chemistry will correspond with experimental results. My time is almost over, so I would like to sum up my talk. We prepared to date about 80 new squaramide analogs, and we performed some advanced tests where we proved the potency, and we want to broaden the SAR study, and we will see how this compound will behave in the future. And we are also working on new derivatives, also compounds designed based on de novo design. And we also prepared about almost 50 novel inhibitors against metalloprotease MP1, and now we are working on another analogs. At the end of my presentation, I would like to thank too many people, especially to my PhD students who synthesized almost all compounds presented today. I would like to thank two colleagues from Belgium who performed all biological tests. I would like to thank my colleague from Palacký University, Professor Šebela, who performed enzymatic assay. Two guys for molecular docking, for financial support, and thank you for your kind attention.
Great, Lucia, thanks very much for that super presentation. Medicinal chemistry is alive and well in the Czech Republic. Well done. So there's two questions coming through now. I don't know the names because they're listed as an anonymous attendee. So the first question was, no cyclobutane ring opening was observed.
No, this is it was nice observation that this cycle is very stable. We tested it against very strong acids like trifluoroacetic acid, and it was really stable under all conditions of what we tried. So, not.
Great. And the second question, which may or may not be the same attendee because it's still anonymous. Any explanation of the reactivity observed with the nucleophilic substitution of chloropyrimidines?
I don't know. It is regarding this one, or can you repeat the question?
Sure. It's any explanation for the reactivity observed with the nucleophilic substitution of chloropyrimidines?
Chloropyrimidines. I think this is to this reaction explanation. Yeah, this is.
It's the regiochemistry, isn't it?
Yeah, about regioisomer. This one is the major one because it's between two nitrogens, and the reaction is faster, and the second one is minor isomer.
Okay, Jakub, you're next for a question, and then I'll go to Basha.
Yeah, I just thank you for the talk, and I just wanted to ask you. I understand you are focused on these two main targets. In the second one, I think it was ZMP1, the metalloprotease. Some of the molecules reminded me of motifs that you would see in so-called PAINs, so PAN-assay.
Yeah.
Sort of interference compounds. So I'm just wondering whether you have some evidence that this might be the case, or whether you look beyond the two targets and selectivity.
Yeah, that's why we started the investigation of indole and pyrrole and indole derivatives, because thiazolidinedione is known to be or belonging between PAINs. So we started with this group a few years ago when I didn't know about this fact, but we got very nice results. But when I found out that this could be very problematic, and it is, because there are now some glitazones that are used in the treatment of diabetes, and now the using of these compounds is stopping. So this could be problematic. And yeah, I know about it, and we would like to avoid to prepare new analogs with diazolidine ring.
Perfect. Thank you.
Thanks, Jakob. And again, the question from Basha is, what is the mode of action for the squareamide analog when it inhibits ATP synthase?
Mode of action is subunit A in ATP synthase. Yeah, it's between the rotating subunit C and yeah, between these two subunits. I don't know if you see the cursor.
Yes.
My mouse. Yeah, it's here, and yeah, it was experimentally proved. We raised continuous mutants when we observed the mutation of one amino acid in the ATPB gene. So it proved that the ATPB gene encode the subunit A. So it was proved. So the exact mode of action is subunit A.
Great, Mikhail.
Yeah, thank you for a nice talk. I just would like to ask about the amino acid fragments you use. Are you using both enantiomers or just L?
Just one. Just one.
Just one.
Yeah. I don't know if I have the time to. It should be here.
Yeah. 34, 35. I was just wondering if there is some effect on DNL amino acid. Oh, okay.
We used optically pure precursors, so we didn't study racemic mixture to compare it. Yeah, but these analogs were not so active as we supposed, so we didn't study it more.
Okay, I see. Thank you.
Great. So maybe we'll leave it at that. Lucia, thanks very much. Excellent presentation, good session of questions and answers. Vladimir, over to you again.
Yeah, thanks. Thanks for a nice lecture. And our last speaker is Petr Bureš from University of Pardubice, and he will talk about organic materials based on cyanocapped molecules. So please, Petr, floor is yours.
Thank you, Vladimir. I will just correct my name. My name is Filip Bureš. I'm coming from the...
Oh, sorry, Bob.
No, no problem. I'm coming from the University of Pardubice, and I will also contribute today a little bit to our highlighting organic chemistry in Czech Republic with my talk entitled cyanocapped organic molecules and their application. So first of all, do you see my slides properly?
Yes.
Okay, super. Change. Okay, perfect. So let's start with the cyano group. Cyano group, it's a well-known functional group or functional derivative of a carboxylic acid, and it constitutes a polarized triple bond with one electron lone pair on the nitrogen atom. And what's most interestingly from the conjugated point of view, that is as linear arrangement, and it possesses negative electronic effects. So generally, it's considered as an electron acceptor, which can be also confirmed by its high Hammett and Petela substitution constant, which are not far from those we know for nitro group. And of course, the increased electronegativity of the carbon atom due to its sp hybridization, it's another supporting feature of the cyano group, which makes the cyano group generally quite interesting in terms of polarizing conjugated systems. As we have also learned from Veronica's lecture, cyano group also possesses peculiar structure and reactivity as compared to other functional derivatives, and it can be also easily followed by infrared spectroscopy. And all those features make cyano group quite popular, not just in organic materials. For today's talk, I have chosen a quartet of cyanocapped molecules. The first one is dicyanoimidazole, which we have applied or generally could be applied as a chromophore, could be applied in variety of sensors, in ion batteries, and similar material-oriented topics. Beside this tripodal molecule, I will focus today also on the triphenylamine NK with cyano group at the periphery, which proved to be also quite efficient analog force for two-photon absorption cross-section. And beside this Y-shaped molecule, we focus also on the X-shaped molecule, which can be represented by dicyanobenzene and dicyanopyrazine, Both can be applied also in nonlinear optics as well as in photoredox catalysis. So let's start with dicyanoimidazole, which in fact it's a general heterocyclic electron acceptor, which we have widely applied in our chemistry. Its synthesis, it's quite easy. If we will start from commercial available diaminomalonitrile, which we have already seen from Veronica's lecture, and its reaction with orthoester, you will get the imidazole, dicyanoimidazole itself. It's also commercial available currently, so you can also buy it. Its bromination and subsequent N-methylation in order to prevent imidazole tautomerism afford quite easily this bromo dicyanoimidazole. And with this moiety in hand, what we have done, it's a couple of cross-coupling reaction, especially Suzuki-Miyaura cross-coupling reaction, in order to generate push-pull type of molecules with dicyanoimidazole as an electron acceptor moiety, various pi-conjugated systems in the middle, and you can have various electron donors at the periphery, as can be also seen from this methoxy biphenyl derivative and its X-ray structure shown here. Beside cross-coupling, we can also done direct nucleophilic substitution. So this is sufficiently reactive also in terms of nucleophilic substitution to get access to the simplest molecule here. And as I already mentioned, those molecules are pretty useful for a variety of reasons as listed here. However, for today, I will focus on pH-induced NLO switch, for which we have utilized these dimethyl amino terminated molecules. And as we have already learned from the Veronica's lecture, this basic moiety, which in fact is also responsible for intramolecular charge transfer and generating APD system, can be easily protonated and deprotonated, which in fact can be reflected by the absorption spectra and the color itself. So generally, upon the protonation, this charge transfer band completely disappeared, and upon the regeneration using triethylmine, you will get this charge transfer band back. So this is what concerns linear optical properties, but we were also interested how does this protonation affect also nonlinear optical properties, especially second order measured by high probability scattering, as you can see from this table in the middle. And as you can see, especially for the sterile and stilbenyl type of compounds with this pi system, we got pretty nice ratio between the protonated and non-protonated protonated form. So generally, we can use them as an NLO switch, whether the protonated form has diminished NLO response, non-protonated one, they are efficient NLO force. The peripheral protonation on dimethyl amino group can be also has also impact on distribution of the frontier molecule orbitals, as shown from those visualization here. Whereas non-protonated form possess charge separation, so both HOMO and LUMO are spread over different part of the molecule. In case of protonated molecules, so we have APA system. In fact, we got the HOMO and LUMO localized over the whole pi conjugated system. This, in fact, B-imidazole with dicyanoimidazole as an electron acceptor, we have also utilized for detection or selective detection of heavy metals, mercury 2 and copper 2 ions in particular. Whereas treatment of this molecule which is originally fluorescent and yellow in the solution with mercury ion caused coordination of the mercury ion to this NC binding pocket, and which result in diminished intermolecular charge transfer. So it's mean decolorization of the solution with persisted emission. In aqueous solution, what we observe, it's in the presence of copper 2 ion. It's copper 2 ion induced hydrolysis of both cyano groups to form this imino-imide moiety, which we call EPIMO. Which is even stronger electron acceptor than both cyano groups present in the original molecule, which is reflected by the bathochromic shift and of course reduced emissive properties. In order to detect both ions easily, we have also developed easy paper strips coated with our imidazole molecule, as can be seen here on this picture. And those paper strips were quite useful for both colorimetric and fluorimetric detection of both ions. even in the complex matrix. Now let me switch to this cyanocape NH imidazole. NH would be quite important to mention at the beginning. And we were interested whether such a molecule would be useful for detection of nerve gases such as sarin or tabun. Of course, we didn't work directly with the tabun or sarin. We work with the mimic, DCP and DCNP as shown here. And when we treated our imidazole with diluted solution of those surrogates, we observe decolorization of the molecule, which is originally yellow, and it's also originally weakly fluorescent. However, upon treatment with DCP or DCNP, the fluorescence increased quite significantly. So we have a pretty nice fluorogenic switch from non-fluorescent state to highly fluorescent blue state. Of course, we are also interested in competitive analytes, whether the sensing of DCP and DCNP is sufficiently selective, which can be demonstrated from this picture here, from which we can easily learn that DCP and DCNP were detected or sensed quite selectively. DCP with the PCL bond has been selected or detected much faster or a little bit faster than DCNP with PCN bond. And of course, we focus also on the mechanism of the sensing, and from phosphorus NMR, we learn easily that upon the treatment of DCP with our imidazole, we got completely new phosphorus signal. So it's mean there must be a new species formed in the reaction mixture. From the DLS, we have also learned that the original large aggregates, which is common for NH imidazole, were destroyed, and we are getting much smaller particles. So it's been from those hints, we propose the following mechanism of sensing, in which the original non-emissive aggregate, upon the treatment with DCP in aqueous media, undergo nucleophilic substitution to afford this N-phosphorylated product, which is in fact highly emissive. So it's mean we were able directly and quickly detect the presence of a nerve gas. And then subsequent time-dependent hydrolysis in the aqueous media take place, affording this non-toxic byproduct and regenerate our original imidazole. So in fact, our simple imidazole behaves as an enzyme, and it's able to detect and also detoxify. And again, in order to make detection of nerve gases easy and undemanding, we have developed a simple paper strip coated with our imidazole, and from the color, which faded proportionally to the concentration of the vapor, we can easily estimate the detection limit of our imidazole, which it's 35 and 42 parts per billion for DCP and DCNP. Now, let me also show you another example of tripodal or Y-shaped molecule, which are generally based on triphenylamine. Triphenylamine is extremely popular central electron donor, widely applied in many chromophores. And its threefold iodination using mercury oxide has been well addressed in the literature, and it's quite easy procedure. And we have done also its partial iodination using iodine and silver nitrate. And then depending on the amount of the iodine, we were able to get this mono and twofold iodinated intermediate. With this threefold iodinated triphenylamine, we were able to construct variety of push-pull molecules, tripodal push-pull molecules by attaching various cyanocape electron-accepting moiety, as listed here on the bottom of the slide. And with such a group of molecules in hand, we were able to nicely tune the emissive properties according the across the visible spectra. We're also able to tune the two-photon absorption properties with some of the examples exceeding, or some of the molecules exceeding 1000 GM unit cross section. partially iodinated triphenyl amines. They were applied for Suzuki Miura cross coupling with this cyano N-cape pi conjugated system or sterile system to get access to linear, quadrupolar, and tripodal molecules. And those molecules we have studied fundamentally because this is quite nice model to study the monochromic, dichromic, and trichromic system and interaction of the particle branches and so on. And from this series of molecules, we have nicely demonstrated that the increasing number of branches of the chromophoric branches doesn't affect the HOMO-LUMO gap. So it's mean a HOMO-LUMO gap in all three molecules is almost the same. However, you can easily tune the optical gap, optical properties, especially the emissive behavior, as well as two photon absorption cross-section. If you will switch to X-shape push-pull molecules, maybe you have already learned that we like alphabet-inspired design of push-pull molecules. And concerning the DCB and DCP moieties, which are generally shown here, and for the construction of such type of push-pull molecules, we needed to develop a synthetic procedure towards this dichlorodicyanobenzene as well as this dichlorodicyanopyrazine. The construction of DCB unit can be done or can be accomplished from phthal imide, which can be iodinated in oleum or diiodinated in the oleum. And the result of this iodination strongly depends on the concentration of the oleum. However, once you get this diode product in hand, you can easily open the imide ring to get this diimide and then perform dehydration in order to generate both cyano group. And this DCP or DPZ moiety has been already shown by Veronica. So this is quite easy synthesis from diaminomalonitrile and its treatment with oxalyl dichloride, leading to this dioxo compound and subsequent chlorination using ternary chloride afforded this dicyanopyrazine. Again, it's currently also commercial available, so you can buy it if you wish. And with both precursor, halogenated precursor in hand, we introduce donor substituted pi linkers via cross-coupling reactions, as shown here, with dimethyl amino as an electron or peripheral electron donor. From the X-ray, you can see how does those molecules look like. They possess generally twisted pi conjugated system, whereas DCP and DPZ moieties are generally planar, which can be also nicely visualized by overlap of the X-ray structure. So we have a planar electron acceptor and twisted dimethyl aniline moiety. From the energy level diagram, we can also compare electron withdrawing ability of both units. The red one, it's for DPZ, the black one for DCB. And you can see where we have almost steady HOMO, which reflects dimethyl amino donor. The red correspond to the dicyanopyrazine acceptor, and the blue numbers correspond, black levels correspond to energy levels of DCB derived compounds. So it's mean by extension of the pi conjugated system, we were also able to tune the HOMO-LUMO gap in both particle series of compounds. And with sufficiently extended pi conjugated system, we got the chromophores with completely separated HOMO and LUMO over two different parts of the molecule. Dicyanopyrazine has been further developed and utilized in photoredox catalysis, which is in fact a modern tool of organic synthesis, already once mentioned here during our lectures. And in fact, it utilizes visible light to generate or initiate some radical transformation in organic synthesis. And this is generally accomplished with the aid of photoredox catalysis. And our DPZ molecule, especially those with methoxythiophene as a substituents at position 5 and 6, proved to be highly active as a photoredox catalyst. So here is the X-ray structure of this molecule, also HOMO and LUMO localization. And as you can see also from this picture, it's orange compound with the absorption maxima or discharge transfer band almost perfectly localized around 450 nanometers, which is exactly the overlap with the emission spectra of white LED. So this is the source widely used for photoredox catalysis. So it's been all those features are very useful to apply apply DPZ as an active photoredox catalyst. And we have subsequently also learned that the DPZ may undergo photo-induced electron transfer. So generally, your DPZ upon the excitation with the light and can be treated with your substrate and perform some redox reaction. And then with the aid of some sacrificial reagent, you can get your catalyst back to a ground state, and you can repeat the whole catalytic cycle. So generally, what you can accomplish in this photo-induced electron transfer is to utilize radicals which you are generating in this part of the cycle, and utilize them, or in fact, to convert the energy of the light into the energy of a chemical bond. And we have proved this ability to undergo a photo-induced electron transfer in variety of reaction, including cross-dehydrogenative coupling, oxidation, hydroxylation, some dehalogenation reduction, some chemodivergent reactions. We have done also some enantioselective olefination and oxidation, as well as some annulation. However, for today's talks, I have chosen something what we have done recently, and it's a photochemical cross-coupling reaction. So generally, it's a reaction in which we generate CC bond in terms of photoredox catalysis. And as a substrate, we have chosen, or model reaction, we have chosen construction of such biheteroaryls. This model reaction, it's the reaction between 4-iodopyridine and pyrrole, and when catalyzed with our catalyst for the irradiation, we have used royal blue ALD. Diisopropyl ethyl amine has been used as a sacrificial reagent, and we got this product with more than 70% yield. In photoredox catalysis, pretty important to perform also some blank experiment, which generally include also the same reaction condition without catalyst. And what we found out that this reaction proceed partially also without presence of the catalyst, which can be explained by oligomerization of the pyrrole upon the irradiation. So you are generating oligopyrroles in the reaction mixture, which can also induce or behave as a weak photoredox catalyst. Then we screen also a variety of radical traps, as shown here. Beside pyrrole, we have also screened N-methyl pyrrole. Again, almost the same result. With diphenyl ethene, this is a really common radical trap, widely used as a benchmark radical trap in photoredox catalysis. We got also this product with and generated this new CC bond, and you can see here exclusively via photoredox protocol. In contrast to this, thiophene, it's the reaction with thiophene, it's much more sluggish, and only in case or in the presence of DPZ, we got just less than 20% of this product. However, this is in accordance with the literature. Thiophene, it's well known that it doesn't behave well as a radical trap in photoredox reactions. Pyridine itself can be also used, and is generally attack at alpha position. So it's mean the reaction with our 4-iodopyridine generated this unsymmetrical bipyridine in 45% yield. We have also screened the positional isomers of the starting iodopyridine, as can learn from those three examples, whereas for 4-iodo and 3-iodopyridine, the reaction worked well. For 2-iodopyridine, the reaction was the reaction outcome or the yield was quite significantly diminished. And when concerned the living group, so it's mean the halogen, the reaction works well with iodo and bromose substituted heteroaromates, as we learn from those two examples, and the reaction outcome is quite diminished when we use the chlorinated products, which is related to the higher bond dissociation energy of this CCl bond. Cosanic diazines, we have screened those two isomers of pyrimidine as shown here, and both work quite commonly and both exclusively via photoredox protocol. So having established the construction of biheteroallyls 6-5, we were also interested in construction of 5-5 biheteroallyls. And for such purposes, we have started from this iodoimidazole, thiazole, and oxazole. And they treatment with pyrrole or N-methyl pyrrole afforded smoothly those 5-membered heteroaryls, which in fact are generally not as easy to construct via convenient metal catalyzed cross-coupling reactions. So having established those fundamental reactivity and scope of the reaction, we were also interested in application of our methodology in more relevant substrate. And the most important one we have screened within the couple of substrate we have screened, it's the coupling between the pyrrole and this 4-fluoroiodobenzene, affording this intermediate, which in fact it's part of the atorvastatin. So it's mean generally we can really easily and almost quantitatively generate this atorvastatin core. Benzothiodiazole or this dibrominated intermediate, it's also quite popular, especially in chemistry of organic materials, conjugated materials as a semiconductors. So we have also screened its ability to be modified via our methodology, and indeed the reaction with pyrrole and N-methyl pyrrole afforded smoothly this unsymmetrically substituted benzothiodiazole. So finally, if you will have a look on the mechanism of the reaction, so generally what we believe that is happening in the reaction mixture, it's excitation of the DPZ catalyst and its subsequent reaction with the diisopropyl ethyl amine to generate a radical anion, which can be excited again to get sufficient energy to to donate electron to the 4-iodopyridine to generate this radical anion, which subsequently upon cleaving this iodide, we are getting the radical, which may radically add to pyrrole, and subsequently with aid of diisopropyl ethyl amine, we are regenerating and re-aromatizing the system of the pyrrole moiety. So finally, let me conclude my talk. I just try to convince you a little bit that cyanocape organic pi conjugated molecules are valuable and useful organic materials with various applications. I have to thank all the group and their involvement in the particular projects which I show you as an assembly today. I also thank all the funding bodies for supporting our research. And last but not least, thank you very much for listening.
Now, Philip, excellent presentation to finish up with. Thank you very much.
Okay.
Smart organic materials makes an impact in Czechoslovakia, there's no doubt. Czech Republic, I should say. Now, over to you, Veronica, for a question.
Thank you for a very nice lecture.
Yep.
Actually, I have to admit that in one of our projects, we were inspired by your synthetic pathways, so we use them. Thank you so much for that. I have a question for your about your imidazole derivatives, imidazole sensors. You presented the increase in fluorescence. Did you determine the quantum yields in on state as well? What is do you know about the value of quantum yields of fluorescence?
You mean here sensing of heavy metals or nerve gases?
Both of them.
Both of them. In fact, no, we didn't care much, you know. So it's like a colorimetric or naked eye detection, which was the most important feature for us, you know.
Okay, I see.
So this is just to have something which really work on the remote areas, just using handheld torch, and that's it. So it's been no any particular equipment you will need to detect them, you know. So it's been this was the goal of both projects.
Thank you for explanation.
Yep.
Thanks.
Welcome.
Thanks, Veronica. And Basha, who certainly wins the prize for most questions asked today. The question is, in DPZ studies, whether the mechanism proceeds through a single electron transfer or a radical cation, radical anion species, and have you confirmed the radical process by EPR studies?
Okay. I will just maybe have a look here. Okay, so generally, in fact, in photoredox catalysis, what you can have oxidative and a reductive quench cycle, and in both cases depends what redox substrate or how you will match your redox or redox property of your substrate to your catalyst. So your catalyst generally may behave as an 1 electron oxidant or 1 electron reductant. So this is the answer to your talk. So it means it's a single electron transfer, but this is the case upon the photoexcitation. We are talking about the excited state of your catalyst, not the ground state.
Great, thank you. And another question in through the Q&A from Vivek Sunderval, who's a postdoc in my group. Hi, Vivek. Thanks, great presentation, Prof. Have you observed any thermally activated delayed fluorescence characteristics using the push-pull fluorophores you have studied?
Yeah, of course. So this is also a well-known study in my group, TADF. It's a methodology widely applied in third generation of OLED materials, which I have also mentioned here. So generally what you need, it's an energetical barrier. So you have one, let's say, moiety of your conjugated system twisted out of the conjugation from the rest of your molecule. And generally upon the excitation, you will get them planarized, and you can consider the TADF mechanism. So this is also the case.
Great, thanks, Philip. Vladimir, you're next up.
Yeah, thank you. I'm just wondering, when you detect these heavy metals, did you also observe some influence of anions?
Yeah, we have done some study, and the impact was Vladimir, I just don't want to fabulate, but I have to check the paper because it's couple of years already.
Right.
But we have studied some chlorides, some halogenides in general, but also some anions with oxidative and reductive properties, and there was not much influence of an anion.
Okay, thank you.
But in fact, we didn't focus on that much, to be honest.
Mm-hmm.
Okay, Veronica.
Sorry, I have one more question.
Yeah, sure.
I'm interested in your push-pull system, but I don't know much about your application. But what are the basic requirements on the acceptoric part? Do you think that it may work with the phthalocyanins, for example?
Yeah, definitely. Yeah, phthalocyanin itself, depending, and you can also tune the properties of phthalocyanin by easy coordination of the particle metals. So it's mean this is another feature. It depends what application are you heading to, you know. Mm-hmm. So if you are going for fluorescent molecules, it's better to have a weak electron acceptor and not really strong ICT. Once you have a molecule with strong ICT, generally you will quench emissive properties. So that's the question, what kind of property, what kind of application are you targeting? And based on this, you have to select the proper pair of donor and acceptor, as well as pi conjugated system.
Okay, thanks.
Thanks, Veronica. Now there's a comment, not a question, in the Q&A from Rosa 2020. Thanks to the organizers and all the speakers for such a wonderful session that allowed us to know more about the organic chemistry advances in the Czech Republic. I didn't write that, by the way. That was Rosa 2020. So there you go.
Okay.
So maybe Philip, we'll pause there if that's okay. And that's the end. Was there any more? Just make sure I didn't miss any. No, I don't think so. Vivek just said thank you for the answer. So Vladimir, would you like to maybe wrap up on behalf of the Czech organic chemistry community, and then I might say a few words at the very end as well.
Sure. First of all, I'm just want to thank you, Pat, and all division of organic chemistry of EuChemS that we were, for this opportunity. I think this is important, and we also, are happy to see that, people around are interested, and we are looking forward to other highlightings because we want to also learn much chemistry around Europe. So thank you.
Great. No, thanks, Vladimir. And again, I learned lots about interesting facts about the history and what's going on in the Czech Republic that I did not know about. And my impression is that it's a really vibrant community and a very important, strong chemical industry. I didn't realize that it had pharmaceutical industries at number 4, and there were such industries as rubber and plastics and chemicals, large-scale chemicals, so that's much bigger. And that's different as we go across Europe. So certainly, I remember from the Irish presentation that the pharmaceutical industry, that was our number 1. So it does change from country to country, and that's interesting to know about it as well. So for all the speakers, Jan, you started off with organocatalysis, making 6-membered rings, 7-membered rings, lots of organocatalysis, a very busy area. Lots of high levels of anti-selectivity. Well done on all of that. Hannah was next, gave us our chemical biology lesson for the day. Very enthusiastic presentation. Lots of hands going there, Hannah. Well done. That's always good to see, even with a Zoom. Because of course, the one thing we don't get from Zoom is people's personality, because you don't see them. But I think we get the vibe from you of how interested you are in those non-canonical RNAs, and finding 9 new examples and where it was all methylated. But really, fundamentally, as chemical biology is so much underpinned by chemical techniques and understanding chemistry to solve those problems. Then we segued to Jakob, who looked at interesting natural product synthesis, gave us 3 examples, lots of spiral compounds, lots of emphasis on spiral compounds in the Czech Republic, I noticed. And then, of course, forskolin and bactobolins, I should say, were the next ones. We then moved on to Veronica, who shed a lot of light, I would say, on azothalocyanins. So interesting presentation there, lots of materials chemistry, which overlapped a little bit, I'd say, with some of Philip's work. So that was interesting to see as well. Of course, in anti-selective synthesis, asymmetric synthesis, we can only measure a percentage EE or even an enantiomeric ratio, depending on how you present it, if you have the chiral columns to separate them. So we had a really interesting presentation there from Mikael on how to be smart and be chiral. That's a good motto, by the way, for the day. So that's our takeaway message for anyone working in asymmetric synthesis. We have to be smart and chiral. But lots of excellent examples about how, the development of chiral columns is not that straightforward, but really you've made it very logical, and a very interesting presentation on that side as well. And then we had a medicinal chemistry from Lucia, lots of examples of antimycobacterials. We saw some squaramides, which are normally only seen in organocatalysis, but here they are showing up as very interesting antimycobacterials. We had pyrimidine analogs, with lots of synthesis, lots of evaluation. And then we finished up just now with Philip again on cyanocap compounds for materials chemistry, nonlinear optical properties and switches, lots of push, lots of pull, lots of synthesis, very interesting presentations. So I'd like to thank all of the speakers for giving up their morning to present on behalf of the Czech Republic. A big thank you to my colleague on the Division of Organic Chemistry Committee, Vladimir Sindelar, who did all the work locally in terms of organizing that local list of speakers, and what great choices he made. And just an advert for the remaining 2 highlighting series this calendar year. In September, it will be highlighting organic chemistry in Portugal, date still to be confirmed. And then in November, and again, a date to be confirmed, we'll be highlighting organic chemistry in the United Kingdom. And we've already got our 4 countries set up for 2024. So it's all rolling away very well. So thank you very much. And with that, I'll sign off. And this has been recorded. You'll see it up on YouTube maybe next week. You can impress your friends by saying that you're up on YouTube. So thanks very much for that. And goodbye from Dublin, and have a nice weekend.
Thank you. Bye-bye.
Thank you.
Bye.
Bye.
Bye-bye.
Bye-bye.